उसका ब्रीथिंग एक्सचेंज, बॉडी फ्लूइड एंड सर्कुलेशन, एक्सक्रीटरी प्रोडक्ट एंड एलिमिनेशन, लोकोमोशन, न्यूरल कंट्रोल एंड कोऑर्डिनेशन एंड केमिकल कोऑर्डिनेशन एंड इंटीग्रेशन। कुछ इतने चैप्टर्स हैं इसमें, जिसमें डाइजेशन एंड एब्जॉर्प्शन चैप्टर से डिलीट हो गया, तो इसको नहीं करेंगे। तो स्टार्ट करने से पहले कुछ छोटा सा अपना कुछ शायरी था, एक यही था कि ये समय सोचने का नहीं, ये समय कुछ करने का है। समय यूं ही बर्बाद नहीं करना, क्योंकि ये समय पढ़ने का है। तो भाई ये समय पढ़ने का है, क्रूशियल टाइम चल रहा है, और उस टाइम कोई भी यार पढ़ते रहो, कुछ ना कुछ तो सीखेंगे ही भाई। हम होंगे कामयाब एक दिन, ये सोच के चलो स्टार्ट करते हैं भाई। यूनिट 5 ह्यूमन फिजियोलॉजी। सो जैसे कि ऑलरेडी मैं बता दूं, इसमें सिर्फ रीडिंग करेंगे, ये सिर्फ रीडिंग वाला सीरीज है, तो इसमें एक्सप्लेनेशन नहीं करेंगे, सिर्फ रीडिंग करेंगे, बस यही एम है इसका। तो साथ में जिसको रीड करना हो भाई बुक खोल के बैठ जाओ, साथ में करते हैं। सो लेट्स गो। अरे भाई, द रिडक्शनिस्ट अप्रोच टू स्टडी of life forms resulted in increasing use of physicochemical concepts and techniques. Majority of these studies employed either surviving tissue model or straightaway cell-free systems. An explosion of knowledge resulted in molecular biology. Molecular physiology became almost synonymous with biochemistry and biophysics. However, it is now being increasingly realized that neither a purely organismic approach nor a purely reductionist molecular approach would reveal the truth about the biological process or living phenomena. Systems biology make us believe that all living phenomena are emergent property due to interaction among components of the system under study. A regulatory network of molecules, supramolecular assemblies, cell, tissue, organisms, and indeed population and communities each create emergent property. In the chapter under this unit, major human physiological processes like digestion, exchange of gases, blood circulation, locomotion, and movement are described in cellular and molecular terms. The last two chapters point to the coordination and regulation body events at the organismic levels. Alfonso Corti, 1822 to 1888. Alfonso Corti, Italian anatomist, was born in 1822. Corti began his scientific career studying the cardiovascular system of reptiles. Later, he turned his attention to the mammalian auditory system. In 1851, he published a paper describing a structure located on basilar membrane of the cochlea containing hair cells that convert sound vibration into nerves impulses, the organ of Corti. He died in year 1988. Chapter 16, Digestion and Absorption. 16.1 Digestive System. 16.2 Digestion of Food. 16.3 Absorption of digestive product. 16 अरे भाई सॉरी, तो डाइजेशन अपन को करना नहीं है भाई, सॉरी, आई एम वेरी सॉरी। इस चैप्टर को स्किप करते हैं। So chapter 17, breathing and exchanges of gases. 17.1 respiratory organs. 17.2 mechanism of breathing. 17.3 exchange of gases. 17.4 transport of gases. 17.5 regulation of respiration. 17.6 disorders of respiratory system. As you have already earlier, oxygen is utilized by organism to directly break down simple molecules like glucose, amino acid, fatty acid, etc. to derive energy to perform various activities. Carbon dioxide, CO2, which is harmful, is also released during the above catabolic reaction. It is therefore evident that oxygen has to be continuously provided to the cell, and carbon dioxide produced by the cell have to be released out. This process of exchange of oxygen from atmosphere with carbon dioxide produced by the cells is called breathing, commonly known as respiration. Please place your hands on your chest. You can feel the chest moving up and down. You know that it is due to breathing. How do we breathe? The respiratory organ that mechanism of breathing are described in the following section of this chapter. 17.1 Respiratory organ. Mechanism of breathing vary among different group of animals depending mainly on their habitats and levels of organization. Lower invertebrates like sponges, coelacanths, flatworms, etc. exchange oxygen to carbon dioxide by simple diffusion over their entire body surface. Earthworms use their moist cuticle, and insects have network of tubes, tracheal tubes, to transport atmospheric air within the body. Special vascularized structure called gills, brachial respiration are used by most of the aquatic arthropods and mollusks, whereas vascularized bag called lungs, pulmonary respiration are used by terrestrial form for the exchange of gases. Among the vertebrates, fishes use gills, whereas amphibians, reptiles, birds, and mammals respire through lungs. Amphibians like frogs can respire through their moist skin, cutaneous respiration also. 70.1.1 Human respiratory system. We have a pair of external nostrils opening out above the upper lips. It leads to the nasal chamber through the nasal passage. The nasal chamber opens into pharynx, a portion of which is the common of passage for food and air. The pharynx opens through the larynx region into the trachea. Larynx is cartilaginous box which help in sound production and hence called sound box. During the swallowing, glottis can be covered by a thin elastic cartilaginous flap called epiglottis to prevent the entry of food into larynx. Trachea is a straight tube extending up to the mid-thoracic cavity, which divides at the level of fifth thoracic vertebra into right and left bronchi. Each bronchi undergo repeated division to form secondary and tertiary bronchi and bronchioles ending up to very thin terminal bronchioles. The trachea, primary, secondary and tertiary bronchi and initial bronchioles are supported by incomplete cartilaginous ring. Each terminal bronchiole gives rise to number of thin, irregular, walled and vascularized bag-like structure called alveoli. The branching network of bronchi or bronchioles and alveoli comprises the lungs. We have two lungs which are covered by a double-layered pleural and pleural fluid between them. It reduces friction on the surface, lung surface. The outer pleural membrane is in close contact with thoracic lining, whereas the inner membrane is in contact with the lung surface. The part starting with the external nostril up to the terminal bronchioles constitute the conducting part, whereas the alveoli and their duct form the respiratory or exchange of gases of the respiratory system. The conducting part transports the atmospheric air to the alveoli. Alveoli clears it from foreign particles, humidifies and also bring the air to body temperature. Exchange part is the site actual diffusion of oxygen and carbon dioxide between blood and atmospheric air. The lungs are situated in thoracic chamber, which is anatomically an airtight chamber. The thoracic chamber is formed by dorsally by the vertebral column, ventrally by sternum, laterally by ribs, and on lower side by dome-shaped diaphragm. The anatomical setup of lungs in thorax is such that any change in volume of the thoracic cavity will reflect in the lungs pulmonary cavity. Such an arrangement is essential for breathing, as we cannot directly alter the pulmonary volume. Respiration involves the following steps. First, breathing of the pulmonary ventilation, by which atmospheric air is drawn in and carbon dioxide rich alveolar air is released out. Second, diffusion of gases, oxygen and carbon dioxide across alveolar membrane. Third, transport of gases by the blood. 4. Diffusion of oxygen and carbon dioxide between blood and tissue. 5th, utilization of oxygen by cells for catabolic reaction and resultant release of carbon dioxide. Cellular respiration is dealt in chapter 14. तो यहाँ पे एक डायग्राम है, इसको 17.4 diagrammatic view of human respiratory system. Sectional view of left lung is also shown. So ये left lung हो गया, right. तो एपिग्लॉटिस ये रहा, लैरिंग्स ये रहा, ट्रैकिया ये रहा, प्लूरल मेम्ब्रेन डबल लेयर ये रहा, एलोलाई Here is pleural fluid between pleural membrane. Here is bronchioles, heart, diaphragm, lung, cord end of ribs एंड ब्रोंकाई. यहाँ पे देखो एक नोच पाया जा रहा है. नोच किस में पाया जा रहा है? थोड़ा सा झुकाव है किस लंग में? ये है लेफ्ट लंग में. तो भाई 17.2 mechanism of breathing. Breathing involves two stages: inspiration, during which atmospheric air is drawn in, and expiration, by which alveolar air is released out. The movement of air into and out of the lung is carried out by creating a pressure gradient between the lungs and atmosphere. Inspiration can occur in the pressure within the lungs. Intrapulmonary pressure is less than the atmospheric pressure. That is, there is a negative pressure in the lung with respect to atmospheric pressure. Similarly, expiration takes place when intrapulmonary pressure is higher than the atmospheric pressure. The diaphragm and specialized set of muscles, external and internal intercostal, between the ribs help in generation of such gradients. Inspiration is initiated by the contraction of diaphragm, which increases the volume of thoracic chamber in anterior-posterior axis. The contraction of external intercostal muscle lift up the ribs and the sternum, causing an increase in the volume of thoracic chamber in dorsoventral axis. The overall increase in thoracic volume cause a similar increase in pulmonary volume. An increase in pulmonary volume decrease the intrapulmonary pressure to less than the atmospheric pressure, which force the air from outside to move into the lungs. That is inspiration. Relaxation of diaphragm and intercostal muscle return the diaphragm and sternum to their normal position and reduce the thoracic volume and thereby pulmonary volume. This led to the increase in intrapulmonary pressure to slightly above the atmospheric pressure, causing an expulsion of air from the lungs. That is expiration. We have ability to increase the strength of inspiration and expiration with the help of additional muscles in the abdomen. On an average, a healthy human breathes 12 to 16 times per minute. The volume of air involved in breathing and movement can be estimated by using spirometer, which help in clinical assessment and pulmonary functions. So here mechanism of breathing. So inspiration A, and second is expiration. Here inspiration, air cavity entering into lung, ribs and sternum rise above, and here is rib cage, diaphragm contract, this come below, volume of thorax increases. Expiration, air expelled out, ribs and sternum return to original position, volume of thorax decreases, diaphragm relaxed and arc upwards. 12.2.1 respiratory volume and capacities. Tidal volume, volume of air inspired or expired during normal respiration. It is approx 500 ml. That is, a healthy man can inspire or expire approximately 6,000 to 8,000 ml of air per minute. Inspiratory reserve volume, IRV, additional volume of air a person can inspire by forceful inspiration. This average 2,500 ml to 3,000 ml. Expiratory reserve volume, additional volume of air a person can expire by forcible expiration. This average 1000 ml to 1100 ml. Residual volume, volume of air remaining in the lung even after a forcible expiration. This average 1100 ml to 1200 ml. By adding up a few respiratory volume described above, one can derive various pulmonary capacities which can be used in clinical diagnosis. Inspiratory capacity, total volume of air a man can inspire after normal expiration. This includes tidal volume and inspiratory reserve volume. TV plus IRV is equal to IC. Expiratory capacity, total volume of air a person can expire after normal inspiration. This includes tidal volume, expiratory reserve volume. EC is equal to tidal volume TV plus ERV. Functional residual capacity, FRC, volume of air that will remain in the lungs after a normal expiration. This includes ERV plus RV. Vital capacity, VC, the maximum volume of air a person can breathe in after a forced expiration. This includes ERV, TV, IRV, or maximum volume of air a person can breathe out after a forced inspiration. Total lung capacity TLC, total volume of air accommodated in the lungs at the end of forced inspiration. This include RV, ERV, TV, and IRV or vital capacity plus residual volume. 70.3 Exchange of gases. Alveoli are primary site of exchange of gases. Exchange of gases also occur between blood and tissue. Oxygen and carbon dioxide are exchanged in this site by simple diffusion, mainly based on pressure concentration gradients. Solubility of the gases as well as thickness of membrane involved in diffusion are also some important factors that can affect the rate of diffusion. Pressure contributed by an individual gas in a mixture of gases is called partial pressure, and it is represented as PO2 for oxygen and PCO2 for carbon dioxide. Partial pressure of these two gases in atmospheric air and two sites of diffusion are given in table 70.1 and in figure 70.3. The data given in the table clearly indicate a concentration gradient for oxygen from alveoli to blood and blood to tissue. Similarly, a gradient is present for carbon dioxide in opposite direction, that is from tissue to blood and blood to alveoli, as solubility of carbon dioxide is 20 to 25% higher than that oxygen. The amount of carbon dioxide that can diffuse through the diffusion membrane per unit in a difference partial pressure is much higher compared to oxygen. The diffusion membrane is made up of three main layers, namely the thin squamous epithelium of alveoli, the endothelium for alveolar capillaries, and the basement substance composed of thin basement membrane supporting the squamous epithelium and the basement membrane surrounding the single layer endothelium as cells of capillaries in between them. However, its total thickness is much less than a millimeter. Therefore, all the factors in our body are favorable for diffusion of O2 from alveoli to tissue and that CO2 from tissue to alveoli. Table 70.1. Partial pressure in mmHg for oxygen and carbon dioxide at different parts involved in diffusion in comparison to those in atmosphere. Respiratory gases, oxygen, carbon dioxide. Atmospheric air, oxygen 51%, in alveoli 104, in deoxygenated blood 40 mmHg, and in blood 95 mmHg, in tissue 40 mmHg. Carbon dioxide in atmospheric air 0.3 mmHg, in alveoli 14 mmHg, in blood 45 mmHg, and in blood oxygenated 40 mmHg, and in tissue 45 mmHg. Figure 17.3. Diagrammatic representation of exchange of gases at alveolus and the body tissue with blood and transport of oxygen and carbon dioxide. Body tissue. So here is inspired air, expired air, and it is alveoli. In alveolar air, PO2 is 104 mmHg, PCO2 is 40 mmHg. After diffusion in pulmonary vein, this is pulmonary vein, and this is systemic artery. In systemic artery, PO2 is 95 mmHg, PCO2 is 40 mmHg. In systemic vein, this vein came from tissue. In tissue, diffusion of oxygen and carbon dioxide take place, and in systemic vein carrying deoxygenated volume, PO2 is 40 mmHg and PCO2 is 45 mmHg, and this is pulmonary artery. Figure 17.4. A diagram of a section of an alveolus with a pulmonary capillaries. Air, squamous epithelium or alveolar wall, one-celled thick. Alveolar cavity, it is basement substance. Endothelium of blood capillary, it is red blood cell, and it is blood capillary. 17.4. Transport of gases. Blood is the medium of for transport of oxygen and carbon dioxide. About 97% of oxygen is transported by RBC in the blood. The remaining 3% of oxygen is carried in dissolved state through the plasma. Nearby 20-25% of carbon dioxide is transported by the RBC, whereas 70% of it is carried as bicarbonate. About 7% of carbon dioxide is carried in dissolved state through plasma. 74.1 transport of oxygen. Hemoglobin is a red-colored iron-containing pigment present in the RBC. Oxygen can bind with the hemoglobin in the reversible manner to form oxyhemoglobin. Each hemoglobin molecule can carry a maximum number of 4 molecules of oxygen. Binding of oxygen with hemoglobin is primarily related to partial pressure of oxygen. Partial pressure of carbon dioxide, hydrogen ion concentration, and temperature are the other factors which can interfere with the binding. A sigmoid curve is obtained when percentage saturation of hemoglobin with oxygen is plotted against the PO2. This curve is called oxygen dissociation curve and is highly useful in studying the effect of factors like PCO2H plus concentration, etc. on binding oxygen with hemoglobin. In the alveoli where there is high PO2, low partial pressure of carbon dioxide, less H plus concentration, and lower temperature, the factors are all favorable for the formation of oxyhemoglobin. Whereas in the tissue where low PO2, high PCO2, high H plus concentration, and higher temperature exist, the condition for favorable for dissociation of oxygen from oxyhemoglobin. This clearly indicates that oxygen gets bound to hemoglobin in lungs surface and get dissociated at the tissue. Every 100 ml of oxygenated blood can deliver around 5 ml of oxygen to the tissue under normal physiological condition. Figure 17.5 oxygen dissociation curve. Partial pressure of oxygen, and here percentage of saturation of hemoglobin with oxygen. In this is sigmoid curve. 17.4.2 transport of carbon dioxide. Carbon dioxide is carried by hemoglobin H carbamino hemoglobin about 20-25%. This binding is related to partial pressure of carbon dioxide. Partial pressure of oxygen is major factor which could affect this binding. When partial pressure of carbon dioxide is high and partial pressure of oxygen is low, as in the tissue, more binding of carbon dioxide occurs, whereas when the partial pressure of carbon dioxide is low and partial PO2 is high, as in alveoli, dissociation of carbon dioxide from carbamino hemoglobin takes place. That is, carbon dioxide which bound to hemoglobin from tissue is delivered at the alveoli. RBC contain a very high concentration of the enzyme carbamino anhydrase, and a minor quantities of same is present in plasma too. This enzyme facilitates the following reaction in both directions. Carbon dioxide plus water. In the presence of carbonic anhydrase is formed H2CO3, and also in presence of carbonic anhydrase, these H2CO3 and dissociated into bicarbonate ion and H plus ion. At the tissue site where partial pressure of carbon dioxide is high due to catabolism, carbon dioxide diffuses into blood, RBC and plasma and form S bicarbonate ion and hydrogen ion. At the alveolar site where PaCO2 is low, the reaction proceeds in the opposite direction, leading to the formation of carbon dioxide and water. Thus, carbon dioxide trapped in bicarbonate at the tissue level and transported to the alveoli is released out as carbon dioxide. Every 100 ml of deoxygenated blood delivers approximately 4 ml of CO2. 17.5 regulation of respiration. Human beings have a significant ability to maintain and moderate the respiratory rhythm to suit the demands of body tissue. This is done by neural system. A specialized center present in medulla region of the brain called respiratory rhythm center is primarily responsible for this regulation. Another center present in the pons region of the brain called pneumotaxic center can moderate the function of the respiratory rhythm center. Neural signal from this center can reduce the duration of inspiration and thereby alter the respiratory rate. A chemosensitive area is situated adjacent to rhythm center, which is highly sensitive to carbon dioxide and hydrogen ion. Increase in these substances can activate this center, which in turn can signal the rhythm center to make necessary adjustment in the respiratory process by which these substances can be eliminated. Receptor associated with aortic arc and carotid artery can also recognize changes in carbon dioxide and H plus concentration and send necessary signal to the rhythm center for remedial action. The role of oxygen in the regulation of respiratory rhythm is quite insignificant. 17.6 Disorders of respiratory systems. Asthma. Asthma is a difficulty of breathing causing wheezing due to inflammation of bronchi and bronchioles. Emphysema is chronic disorder in which alveolar wall are damaged, due to which respiratory surface is decreased. One of the major causes of this is cigarette smoking. Occupational respiratory disorders. In certain industries, especially those involving grinding of or stone breaking, so much dust is produced that the defense mechanism of the body cannot fully cope with this situation. Long exposure that give rise into inflammation, leading to fibrosis, proliferation of fibrous tissue, and thus causing serious lung damage. Workers in such industries should wear protective masks. Summary. Cell utilize oxygen for metabolism and produce energy along with substances like carbon dioxide, which is harmful. Animals have evolved different mechanisms for transport of oxygen to the cells and for removal of carbon dioxide from there. We have a well-developed respiratory system comprising of two lungs and associated their passage to perform these functions. The first step in respiration is breathing, by which atmospheric air is taken in inspiration and alveolar air is released out, expiration. Exchange of oxygen and carbon dioxide between deoxygenated blood and alveoli, transport of these gases throughout the body by blood, exchange of oxygen and carbon dioxide between oxygenated blood and tissue, and utilization of carbon dioxide by the cells. Cellular respiration are the other steps involved. Inspiration and expiration are carried out by creating pressure gradients between the atmosphere and alveoli with the help of specialized muscles, intercostals and diaphragm. Volume of air involved in these activities can be estimated with the help of spirometer and are clinical significance. Exchange of oxygen and carbon dioxide at alveolar tissue occur by diffusion. Rate of diffusion is dependent on partial pressure gradient of oxygen PO2 and CO2 PCO2, their solubility as well as the thickness of diffusion surface. These factors in our body facilitate diffusion of oxygen from alveoli to the deoxygenated blood as well as from oxygenated blood to tissue. The factors are favorable for diffusion of carbon dioxide in opposite direction, that is from tissue to alveoli. Oxygen is transported mainly in as oxyhemoglobin in the alveoli, where PO2 is higher and oxygen get bound to hemoglobin, which easily dissociate at the tissue, where partial pressure of oxygen is low and partial pressure of carbon dioxide and H plus are high. Nearly 70% of carbon dioxide is transported as bicarbonate ion with the help of enzyme carbonic anhydrase. 20 to 25% of carbon dioxide is carried by hemoglobin as carbaminohemoglobin. In the tissue where partial pressure of carbon dioxide is high, it gets bound to blood, whereas in the alveoli where partial pressure of carbon dioxide is low and partial pressure of oxygen is high, it gets removed from the blood. Respiratory rhythm center is maintained by respiratory center in medulla region of the brain, a pneumotoxic center in the pons region of the brain, and a chemosensitive area in medulla can alter respiratory mechanism. So मेरे भाई Chapter 18, Blood Fluid and Circulation, 2:47 to 2:51 PM ब्रेक रहेगा मेरे भाई। इसके बाद स्टार्ट करेंगे अपन बॉडी फ्लूइड एंड सर्कुलेशन। अच्छा चलता हूँ, दुआओं में याद रखना मेरे जिक्र का। मेरे रंग में रंगने वाली परी हो या हो परियों की रानी, या हो कोई प्रेम कहानी। और भाई क्या हालचाल। चलो ब्रेक है थोड़ा, कुछ शायरी देख लेते हैं भाई लोगों के लिए। मेरे रंग में रंगने वाली परी हो या कोई परियों की रानी, तो है टाइम हो गया, रेस्ट हो गया ना भाई लोग। लेट्स स्टार्ट न्यू चैप्टर बॉडी फ्लूइड एंड सर्कुलेशन। तो इसमें पढ़ेंगे 18.1 ब्लड, 18.2 लिम्फ टिश्यू फ्लूइड, 18.3 सर्कुलेटरी पाथवे, 18.4 डबल सर्कुलेशन, 18.5 रेगुलेशन ऑफ कार्डियक एक्टिविटी, 18.6 डिसऑर्डर ऑफ सर्कुलेटरी सिस्टम। तो you have learned that all living cells have to be provided with nutrient, oxygen and other essential substances. Also, the waste of harmful substances produced have to be removed continuously for healthy functioning of tissue. It is therefore essential to have efficient mechanism for the movement of these substances to the cells and from the cells. Different group of animals have evolved different method for this transport, and simple organisms like sponges and coelenterate circulate water from their surrounding through their body cavities to facilitate cells to exchange these substances. More complex organisms use special fluid within their bodies and transport such market material materials. Blood is most commonly used body fluid by most of most of the higher organisms, including humans, for this purpose. Another body fluid, lymph, also help in the transport of certain substances. In this chapter, you will learn about the composition and properties of blood and lymph tissue, and the mechanism of circulation of blood is also explained here. 18.1 Blood. Blood is special connective tissue consisting of fluid matrix, plasma, and foreign element. 18.1.1 Plasma. Plasma is straw colored viscous fluid constituting nearly 55% of blood. 90 to 95% of plasma is water and protein contribute 6 to 8% of it. Fibrinogen, globulin, and albumin are major proteins. Fibrinogen are needed for clotting of or coagulation of blood. Globulins primarily involved in defense mechanism of the body, and albumin help in osmotic balance. Plasma also contain a small amount of minerals like sodium ion, calcium ion, magnesium ion, bicarbonate ion, chloride ion, etc. Glucose, amino acid, lipid, etc. are also present in the plasma as they are always transit in body. Factors for coagulation or clotting of blood are also present in plasma in an inactive form. Plasma without clotting factor is called serum. 18.1.2 Formed elements. Erythrocytes, leukocytes, and platelets are collectively called formed elements, and they constitute nearly 45% of blood. Erythrocytes or red blood cells (RBC) are most abundant of all the cell in body. A healthy adult man has on an average 5 million to 5.5 million of RBC per millimeter cube of blood. RBC are formed in red bone marrow in adults. RBC are devoid of nucleus in most of the mammals and are biconcave in shape. They have red colored iron containing complex protein called hemoglobin. Hence the color and name of these cells. A healthy individual has 12 to 16 gram of hemoglobin in every 100 ml of blood. These molecules play a significant role in transport of respiratory gases. RBC have an average life span of 120 days, after which they are destroyed in spleen, graveyard of RBC. Leukocytes. Leukocytes are also known as white blood cells (WBC) as they are colorless due to lack of hemoglobin. They are nucleated and are relatively lesser in number, which average 6,000 to 8,000 per mm³ of the blood. Leukocytes are generally short-lived. We have two main categories of WBC: granulocytes and agranulocytes. Neutrophils, eosinophils, and basophils are different types of granulocytes, while lymphocytes and monocytes are agranulocytes. Neutrophils are most abundant cells, 60 to 65% of the total WBC, and basophils are the least, 0.5 to 1% among them. Neutrophils and monocytes, 6 to 8%, are phagocytic cells which destroy foreign organisms entering in the body. Basophils secrete histamine, serotonin, heparin, etc. and are involved in inflammatory reaction. Eosinophils, 2 to 3%, resist infections and are also associated with allergic reaction. Lymphocytes, 20 to 25%, are of two major types, B and T form. Both B and T lymphocytes are responsible for immune responses of the body. Platelets are also called thrombocytes, are cell fragments produced from megakaryocytes, special cell in the bone marrow. Blood normally contains 150,000 to 350,000 platelets per mm³. Platelets can release a variety of substances, most of which are involved in the coagulation of or clotting of blood. or reduction in the number of can lead to clotting disorder, which will lead to excessive loss of blood from body. 18.1.3 Blood groups. As you know, blood of humans being differ in certain aspects, though it appear to be similar. Various type of group of the body has been done to such grouping. The ABO and RH are widely used all over the world. 18.1.1 18.1.3.1 ABO grouping. ABO grouping is based on the presence and absence of two surface antigens, chemical that can be induced immune responses on the RBC, namely or A and B. Similarly, the plasma of different individuals contain two natural antibodies, proteins produced in response of antigens. The distribution of antigen and antibodies in the four group of blood A, B, AB, and O are given in table 18.1. You probably know that during blood transfusion, any blood cannot be used. The blood of donor has to be carefully matched with the blood of recipient before any blood transfusion to avoid severe problems of clumping, destruction of RBC. The donor compatibility is also shown in figure 18.1. For the above mentioned table, it is evident that oxygen blood can be donated to a person with any other blood groups, and hence oxygen group O group individual are called universal donors. Person with AB group can accept blood from person with AB as well as other groups of blood. Therefore, such person called universal recipient. Here 18.1 diagram representation of formed element in the blood. RBC, platelets, eosinophils, basophils, neutrophil, monocytes, T lymphocytes, B lymphocyte. Table 18.1 blood grouping and donor compatibility. Blood group A, antigen or BC, A, antibodies in plasma, anti-B. Donor group A and O. Blood group B, antigens on RBC, B, antibodies in plasma, anti-A. Donor group B and O. Blood group AB, antigen on RBC, A or B, antibodies in plasma, nil. Donor group AB, A, B or O. Blood group O, antigen no antigen, nil. Antibodies anti-A or B. Donor group oxygen. 18.1.3.2 RH grouping. Another antigens. The RH antigen similarly to one present in rhesus monkey, hence RH, is also absorbed on the surface RBC of majority, nearly 80% of humans. Such individuals are called RH positive. RH positive, and those in whom this antigen is absent are called RH negative. RH negative. RH negative, if exposed to RH positive blood, will form specific antibodies against RH antigens. Therefore, RH groups should also be matched before transfusion. A special case of RH incompatibility mismatching has been observed between RH negative blood of pregnant mother with RH positive blood of the fetus. RH antigen of fetus do not get exposed to RH negative blood of mother in the first pregnancy, as the two bloods are well separated by placenta. However, during the delivery of the first child, there is a possibility of exposure of maternal blood to a small amount to RH positive blood from fetus. In such cases, the mother starts preparing antibodies against RH antigen in her blood. In case of subsequent pregnancy, the RH antibody from the mother RH negative can leak into the blood of fetus RH positive and destroy the fetal RBC. This could be fatal to the fetus or cause severe anemia and jaundice to the baby. This condition is called erythroblastosis fetalis. This can be avoided by administering anti-RH antibodies to the mother immediately after delivering the first child. 18.1 Coagulation of blood. You know that when you cut your finger or hurt yourself, you does not continue to bleed for a long time. Usually the blood stops flowing after some time. Do you know why? Blood exhibits coagulation or clotting in response to an injury or trauma. This is a mechanism to prevent excessive loss of blood from the body. You would have observed a dark reddish brown scum formed at the site of cut or an injury over a period of time. It is a it is a clot or coagulum formed mainly of a network of threads called fibrin, in which dead and damaged formed elements of blood are trapped. Fibrins are formed by conversion of inactive fibrinogens in the plasma by enzyme thrombin. Thrombins in are found from another inactive substances present in the plasma called prothrombin. An enzyme complex thrombokinase is required for above reaction. This complex is formed by a series of linked enzyme reaction cascade process involving a number of factors present in the plasma in an inactive state. An injury or trauma stimulates the platelets in blood to release certain factors which activate the mechanism of coagulation. Certain factors released by tissue at the site of injury also can initiate coagulation. Calcium ion play a very important role in clotting. 18.2 Lymph tissue fluid. As the blood passes through the capillaries in tissue, some water along with many small water-soluble substances move out into the spaces between the cells of the tissue, leaving the larger protein and most of the formed element in the blood vessels. This fluid released out in is called the interstitial fluid or tissue fluid. It has the same mineral distribution as that in plasma. Exchange of nutrients, gases, etc. between the blood and the cells always occur through this fluid. An elaborate network of vessels called lymphatic system collect this fluid and drains it back to the major veins. The fluid present in the lymphatic system is called lymph. Lymph is colorless fluid containing specialized lymphocytes which are responsible for immune responses of the body. Lymph is also an important carrier of nutrients, hormones, etc. Fat are absorbed through lymph in the lacteals present in the intestinal villi. 18.3 Circulatory pathway. The circulatory pattern are two types, open and closed. Open circulatory system is present in arthropod and mollusk, in which blood pumped by heart passes through the large vessels into open spaces or body cavities called sinuses. Annelids and chordates have a closed circulatory system in which the blood pumped by the heart is always circulated through a closed network of blood vessels. This pattern is considered to be more advantageous as the flow of fluid can be more precisely regulated. All vertebrates possess a muscular chambered heart. Fishes have a two-chambered heart with an atrium and ventricular. Amphibians are the reptiles except crocodile have three-chambered heart with two arteries and a single ventricle, whereas crocodiles, birds, and mammals possess a four-chambered heart with two arteries and two ventricles. In fishes, the heart pumped out deoxygenated blood, which is oxygenated by gills and supplied to the body parts, from where deoxygenated blood is returned to the heart. Single circulation. In amphibians and reptiles, the left atrium receives oxygenated blood from gills, lungs, skin, and the right atrium gets deoxygenated blood from other body parts. However, they get mixed up in a single ventricle, which pumps out mixed blood in complete double circulation. In birds and mammals, oxygenated and deoxygenated blood received by left and right atria respectively pass onto the ventricles of same side. The ventricles pumps it out without any mixing. Two separated circulatory pathways are present in these organisms. Hence, these animals have double circulation. Let us study human circulatory system. 18.3.1 Human circulatory system. Human circulatory system, also called blood vascular system, consists of muscular chamber heart, a network of closed branching blood vessels, and blood, the fluid which is circulated. Heart. Heart is mesodermally derived organ, is situated in the thoracic cavity in between the two lungs, slightly tilted to the left. It has the size of clenched fist. It is protected by a double-walled membranous bag pericardium enclosing the pericardial fluid. Our heart has four chambers, two relatively small upper chambers called atria and two larger lower chambers called ventricles. A thin muscular wall called interarterial septum separates the right and left atria, whereas a thick walled interventricular septum separates the left and right ventricles in figure 18.2. The atrium and ventricles of the same side are also separated by thick fibrous tissue called atrioventricular septum. However, each of these septa are provided with opening through which the two chambers of the same side are connected. The opening between the right atrium and right ventricle is guarded by a valve formed of three muscular flaps or cusps called tricuspid valve, whereas the bicuspid or mitral valve guarded by opening between right and left ventricle. Pulmonary artery and aorta respectively are provided with the semilunar valve. The valve in the heart allows the blood only in one direction, that is from artery to ventricle and from ventricle to the pulmonary artery or aorta. These valves prevent any backward flow. The entire heart is made of cardiac muscles. The wall of ventricles are much thicker than that of arteria. A specialized cardiac musculature called nodal tissue is also distributed in the heart. A patches of tissue present in right upper corner of right atrium called sinoatrial node, SA node. Another mass of this tissue is seen in lower left corner of right atrium close to atrial ventricular septum called AV node. A bundle of nodal fibers atrioventricular bundle AV bundle continues from AV node, which passes through atrioventricular septa to emerge on the top of interventricular septum and immediately divide into right and left bundle. These branches give rise to minor fibers throughout the ventricular musculature of respective sides and are called Purkinje fibers. The nodal musculature has the ability to generate action potential without any external stimuli. That is, it is auto-excitable. However, the number of action potential that could be generated in minute vary at different parts of the nodal system. The SN node can generate maximum number of action potential, that is from 70 to 75 per minute, and is responsible for initiating and maintaining the rhythmic contractile activity of the heart. Therefore, it is called pacemaker. Our heart normally beats 70 to 75 times a minute, average 72 beats per minute. Figure 18.2, section of human heart. Aorta, pulmonary artery, pulmonary vein, left atrium, bundle of His, left ventricle, and it is ventricular septum, and it is heart apex, right atrium, cordia tendini, and this is AV node, and this is right atrium, this is SA node, and this is vena cava. 18.3.2 cardiac cycle. How does the heart function? Let us take a look. To begin with, all the four chambers of heart are relaxed state. They are in joint diastole. As the tricuspid and bicuspid valve are open, blood from the pulmonary vein and vena cava can flow into the left and the right ventricles, respectively, through the left and right atria. The semilunar valve are closed at this stage. The SA node now generate an action potential which stimulate both the atria to undergo simultaneously contraction the atrial systole. This increases the flow of blood into ventricles by about 30%. The action potential is conducted to the ventricular side by AV node and AV bundle, from where the bundle of heat transmits it through the entire ventricular musculature. This causes the ventricular muscle to contract, ventricular systole. The atria undergo relaxation diastole coinciding with ventricular systole. Ventricular systole increase the ventricular pressure, causing the closure of tricuspid and bicuspid valve due to attempt backflow of blood into atria. As the ventricular pressure increases further, the semilunar valve guarding the pulmonary artery right side and aorta left side are forced open, allowing the blood in the ventricle to flow through this vessel into circulatory pathway. The ventricles now relax ventricular diastole and the ventricular pressure falls, causing the closure of semilunar valve, which prevents the backflow of blood into ventricles. As the ventricular pressure declines further, the tricuspid and bicuspid valve are pushed open by the pressure of arteria exerted by the blood which was being emptied into them by veins. The blood now once again moves freely to the ventricles. The ventricles and arteria are now again in relaxed joint diastole state as earlier. Soon the SN node generates a new action potential and event described above are repeated in that sequence and the process continues. The sequential event in the heart which is cyclic repeated is called cardiac cycle and it consists of systole and diastole, both atria and ventricles. As mentioned earlier, the heart beats 72 times per minute. That is that many cardiac cycles are performed per minute. From this, it could be deduced that the duration of cardiac cycle is 0.8 seconds. During a cardiac cycle, each ventricle pumped out approximately 70 ml of blood, which is called stroke volume. The stroke volume multiplied by the heart rate, no number of beats per minute, give to cardiac output. Therefore, the cardiac output can be defined as volume of blood pumped out by every ventricle per minute and average 5000 ml or 500 liter in healthy individual. The body has the ability to alter stroke volume as well as heart rate by cardiac output. For example, the cardiac output of an athlete will be much higher than the ordinary man. During each cardiac cycle, two prominent sound produced, which can be easily heard through stethoscopes. The first heart sound love is associated with the closure of tricuspid and bicuspid valve, whereas the second heart sound is dub associated with the closure of the semilunar valve. This sound are clinical diagnostic significant. 18.3.3 Electrocardiograph or ECG. You are probably familiar with these scenes from a typical hospital television shows. A patient is hooked up to the monitoring machine that shows voltage traces on screen, make up the sound beep 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 as the patient goes into the cardiac arrest. This type of machine, electrocardiograph, is used to obtain electrocardiogram. ECG. ECG is graphical representation of electrical activity of heart during cardiac cycle. To obtain a standard ECG as shown in the figure 18.3, a patient is connected to the machine with 3 electrodes, one on each wrist and one to left ankle, that continuously monitor the heart activity. For a detailed evaluation of heart function, multiply lead to the attached to chest region. Here, we will talk about standard ECG. Each peak of ECG is identified with a letter P to T that correspond to the specific electrical activity. The P wave represents the electrical excitation or depolarization of the arteria, which led to the contraction of both the arteria. The QRX complex represents the depolarization of the ventricles, which initiate the ventricular contraction. The contraction starts shortly after Q and marks the beginning of the systole. The T wave represent the return of ventricles from excited to normal state repolarization. The end of T waves marks the end of systole. Obviously, by counting the number of QRS complex that occur in a given time periods, one can determine the heartbeat rate in individual. Since ECG obtained from different individuals have roughly the same shape given led to the configuration, any deviation from this shape indicate a possible abnormality or disease. Hence, it is great clinical significance. 18.3 Diagrammatic representation of standard ECG. Here P wave, QRAS and T wave. This represent a depolarization of arteria. This represent depolarization of ventricle, and T wave represent repolarization of ventricle. 18.4 double circulation. The blood flows strictly by a fixed route through blood vessels, the artery and veins. Basically, each artery and vein consists of three layers and inner lining of squamous endothelium. The tunica intima, a middle layer of smooth muscle and elastic fiber tunica media, and an external fibrous connective tissue with collagen fiber tunica externa. The tunica media is comparatively thin in the veins. As mentioned earlier, the blood pumped by the right ventricles enters the pulmonary artery, whereas the left ventricle pumps blood into the aorta. The deoxygenated blood pumped into the pulmonary artery is passed on to the lungs, from where the deoxygenated blood is carried by the pulmonary veins into the left atrium. This pathway constitutes the pulmonary circulation. The oxygenated blood entering the aorta is carried out by a network of arterioles and capillaries to the tissue, from where deoxygenated blood is collected by a system of venules, veins, and vena cava and emptied into the right atrium. This is the systemic circulation. The systematic circulation provides nutrients, oxygen, and other essential substances to the tissue, take carbon dioxide and other harmful substances away for elimination. A unique vascular connection exists between digestive tract and liver called hepatic portal system. The hepatic portal in veins carries blood from intestine to the liver before it delivered to the systemic circulation. A special coronary system of blood vessel is present in our body exclusively for circulation of blood and from cardiac musculature. 18.4 schematic plan for blood circulation in humans. Lungs, this is pulmonary vein, this is aorta, and this is artery. You can see it is more thicker than this vein. Is smooth muscle, and this is lumen. This is capillary, and this is vein, smooth muscle, and lumen is broader than that present in artery. This is vena cava, great vein, and this is heart, right atrium, right ventricle, left atrium, left ventricle, and this is pulmonary artery. 18.5 regulation of cardiac activity. Normal activity of heart is regulated intrinsically, that is auto-regulated by a specialized muscle nodal tissue. Hence, the heart is called myogenic. A special neural center in medulla oblongata can moderate the cardiac function through autonomic nervous system (ANS). Neural signals through sympathetic nerves part of ANS can increase the rate of heartbeat, the strength of ventricular contraction, and thereby the cardiac output. On the other hand, parasympathetic neural signals, another component of ANS, decrease the rate of heartbeat, speed of conduction of action potential, and thereby the cardiac output. Adrenal metabolic hormone can also increase the cardiac output. 18.6 disorder of circulatory system. High blood pressure, hypertension. Hypertension is the term for blood pressure that is higher than normal human, 120 to 280. In this measurement, 120 is in mmHg, millimeter of mercury pressure, is the systolic or pumping pressure, and 80 mmHg is diastolic or resting pressure. If repeated check of blood pressure of an individual is 140 to 90, 140 over 90, or what is this bro? Sorry भाई लोग, क्या क्या हो जाता है ब्रो. मेरे रंग में रंगने वाली भाई सॉरी ब्रो, सबको बहुत बहुत सॉरी. क्या है कि मेरे इस डिवाइस में क्या है कि स्टोरेज बहुत ज़्यादा फुल हो चुका है, और सारे पीडीएफ से इतने पीडीएफ रखा हूँ इसमें, इतने पीडीएफ हैं ब्रो, इतने. व्हाट इज दिस ब्रो. हम्म, मेरे रंग में रंगने वाली परी हो या हो परियों की रानी। ब्रो वेरी वेरी सॉरी भाई लोग आपको, आई एम वेरी वेरी सॉरी ब्रो। कहाँ तक किए थे भाई लोग? क्या करो यार, स्टोरेज फुल हो गया। डिवाइस का गलती नहीं है, मेरा गलती है। इतना इसको लोड दे दिया हूँ, बच्चा इतने फाइल भर दिए इसमें कि बेचारा कंट्रोल नहीं हो कर पा रहे। आई एम सॉरी भाई, जितने भी दोस्त हैं देख रहे हैं वीडियो को, बहुत बहुत सॉरी ब्रो। 18.5 रेगुलेशन ऑफ ब्लड एंड कार्डिक। मेरे रंग में 18.3 regulation of cardiac activity. Normal activity of heart are regulated intrinsically, that is auto-regulated by specialized muscle neural tissue. Hence the heart is called myogenic. A special neural center in medulla oblongata can moderate the cardiac function through autonomic nervous system ANS. Neural signal through a sympathetic nerves part of ANS can increase the rate of heartbeat, the strength of ventricular contraction, and thereby the cardiac output. On the other hand, the parasympathetic neural signal, another component of ANS, decrease the rate of heartbeat, speed of conduction of action potential, and thereby cardiac output. Adrenomedullary hormone can increase the cardiac output. 18.6 disorder of circulatory system. High blood pressure, hypertension. Hypertension is the term of blood pressure that is higher than normal, 120 to 80. In this measurement, 120 mmHg millimeter of mercury pressure is systolic or pumping pressure, and 80 mmHg is diastolic or resting pressure. If repeated, check the blood pressure of individual 140 to 90, 140 over 90 or higher. It's hypertension. High blood pressure lead to the heart disease and also affect vital organs like blood, brain, and kidney. Coronary artery disease (CAD). Coronary artery disease is often referred to as atherosclerosis. Affect the vessel that supply blood to the heart muscle. It is caused by deposits of calcium, fat, cholesterol, and fibrous tissue, which make the lumen of artery narrower. Angina. It is also called angina pectoris, or symptoms of acute chest pain which occur when no enough oxygen is reaching to the heart muscle. Angina can occur in men and women of any ages, but it is also common among the middle-aged and elderly. It occurs due to the condition that affect the blood flow. Heart failure. Failure means the state of heart when it's not pumping blood effectively enough to meet the needs of body. It is sometimes called congestive heart failure because of congestion of the lung is one of the main symptoms of this disease. Heart failure is not the same as cardiac arrest when the heart stops beating, or heart attack when the heart muscle suddenly damaged by inadequate supply of blood supply. Summary: vertebrates circulate blood, a fluid connective tissue, in their body to transport essential substances to the cells and carry waste substances from there. Another fluid lymph tissue is also used for transport of certain substances. Blood comprises of a fluid matrix, plasma, and formed element. Red blood cells, RBC, erythrocytes, white blood cells, WBC, leukocytes, and platelets, thrombocytes constitute the formed elements. Blood of humans are grouped into A and B and O system based on the presence or absence of two surface antigens, A and B on RBC. Another blood grouping is also done based on the presence or absence of another antigens called rhesus factor, RH, on the surface of RBC. The spaces between cells in tissue contain a fluid derived from blood called tissue fluid. This fluid is called lymph, is almost similar to the blood except for protein content and formed elements. All vertebrate and a few vertebrate have closed circulatory system. Our circulatory system consists of muscular pumping organ, heart, a network of vessels, and a fluid and blood. Heart has two arteries and ventricles. Cardiac musculature is auto-excitable. SA node generate maximum number of action potential per minute, 70 to 75 minute per minute, and therefore it sets the pace of activity of heart. Hence, it is called pacemaker. The action potential causes the atria and ventricles to undergo contraction systole, followed by their relaxation diastole. The systole forces the blood to move from atria to ventricles and the pulmonary artery and the aorta. The cardiac cycle is formed by sequential event in the heart, which is cyclically repeated and is called cardiac cycle. A healthy person shows 72 such cycles per minute. About 70 ml of blood pumped out of each ventricle during a cardiac cycle, and it is called stroke or beat volume. Volume of blood pumped out by each ventricle of heart per minute is called the cardiac output, and it is equal to product of stroke volume and heart rate, approx 5 liters. The electrical activity of heart can be recorded from the body surface by using electrocardiograph, and recording is called electrocardiogram (ECG), which is clinical importance. We have a complete double circulation, that is two circulatory pathway, namely pulmonary and systemic are present. The pulmonary circulation starts by the pumping of deoxygenated blood by the right ventricle, which is carried to the lungs. When it is oxygenated and returned to the left atrium, the systemic circulation starts with pumping of oxygenated blood by the left ventricle to the aorta, which is carried to all the body tissue, and the deoxygenated blood from there is collected by veins and returned to right atrium. Though the heart is auto-excitable, its function can be moderated by neural and hormonal mechanism. तो दो चैप्टर कंप्लीट हुआ भाई ह्यूमन फिजियो का, ब्रीथिंग, एक्सचेंज ऑफ गैस एंड सर्कुलेशन. अब चैप्टर 90 करेंगे अपन, एक्सक्रीटरी प्रोडक्ट एंड देयर एलिमिनेशन, चैप्टर 90. तो 3:30 हो रहा है, 3:33 PM तक अपन का रहेगा ब्रेक. उसके बाद करते हैं ब्रो स्टार्ट. मेरे रंग में रंगने वाली. चलो भाई मिलते हैं 3 मिनट बाद, कहीं नहीं जाना. तो भाई अभी 2 मिनट है, कुछ चर्चा करा जाए क्या? ब्रेक का 2 मिनट है, तो इस पे एक कुछ हो जाए क्या, स्वैग वाली बात. अगर नहीं पढ़ा है जो भाई अभी तक, उसके लिए शायरी है, और जो पढ़ा है उसके लिए, सबके लिए बढ़िया शायरी. तो कुछ ऐसे है, थोड़ा सब्र रखो हम भी मैदान में आएंगे, और माहौल क्या है होता है वो भी बताएंगे. तो भाई मैदान में उतरे ही नहीं थे, लोग समझ रहे थे हम पढ़ते ही नहीं है। अब उतरे हैं भाई, दिखा दो अपना जलवा, तोड़ डालो, फोड़ डालो। जब तक नहीं किए नहीं किए, जब करना स्टार्ट किए तो कोई आगे दिखना नहीं चाहिए ब्रो, ठीक है। करो भाई मेहनत खतरनाक, दिखा दो सबकी उनकी क्या है औकात। लेट्स गो ब्रो। परी हो या कोई परियों की रानी, क्या हाल। अभी 33 करना है ब्रो। अभी भाई अपने पास 1 मिनट है, उसके पहले अपन स्टार्ट नहीं कर सकते हैं इसको। तो और बताओ भाई, कैसा चल रहा है तुम लोग का पढ़ाई? हो गया ब्रो टाइम, लेट्स गो। तो इसमें कुछ एक्सक्रीटिव प्रोडक्ट एंड एलिमिनेशन क्या पढ़ेंगे। 90.1 ह्यूमन एक्सक्रीटिव सिस्टम, 90.2 यूरिन फॉर्मेशन, 90.3 फंक्शन ऑफ ट्यूब्यूल्स, 90.4 मैकेनिज्म ऑफ concentration of the filtrate, 90.5 regulation of kidney function, 90.6 micturition, 90.7 role of other organs in excretion, 90.8 disorders of excretory system. Animals accumulating ammonia, urea, uric acid, carbon dioxide, water, and ions like sodium, potassium, chloride, iron, phosphate, sulfate, etc., either by metabolic activities or by other means like excess ingestion. These substances have to removed totally or partially. In this chapter, you will learn the mechanism of elimination of these substances with special emphasis on common nitrogenous waste. Ammonia, urea, and uric acid are the major form of nitrogenous waste excreted by the animals. Ammonia is the most toxic form and require large amount of water for their elimination, whereas uric acid, being the least toxic, can be removed with a minimum loss of water. The process of excreting ammonia is called ammonotelium. Many bony fishes, aquatic amphibians, and aquatic insects are ammotelic in nature. Ammonia, as it is readily soluble, is generally excreted by diffusion across body surface or through gill surface in fish as ammonium ions. Kidneys do not play any significant role in its removal. Terrestrial adaptation necessitated the production of lesser toxic nitrogenous waste like urea and uric acid for conservation of water. Mammals, many terrestrial and amphibians are marine fishes, mainly excrete urea and called ureotelic animals. Ammonia produced by metabolism and converted into urea in the liver. These animals and released into blood, which is filtered and excreted out by the kidney. Some amount of urea may be retained in the kidney matrix of some of these animals to maintain a desired osmolarity. Reptiles, birds, land snails, and insects excrete ammonia waste as uric acid in the form of pellets or pastes with minimum loss of water and are called uricotelic animals. A survey of animal kingdom presents a variety of excretory structures. In most of invertebrates, these structures are simple tubular form, whereas vertebrates have a complex tubular form called kidney. Some of these structures are mentioned here. Protonephridia or flame cells are excretory structure in platyhelminthes flatworm, that is planaria, rotifers, some annelids, and cephalochordate. Amphioxus. Protonephridia are primarily concerned with ionic and fluid volume regulation, that is osmoregulation. Nephridia are tubular excretory structure of earthworm and other annelids. Nephridia help to remove nitrogenous waste and maintain a fluid and ionic balance. Malphizian tubules and excretory structure of most of the insects, including cockroaches. Malphizian tubules help in the removal of nitrogenous waste and osmoregulation. Antennal gland or green gland perform the excretory function in crustaceans like prawns. 19.1 Humans excretory system. In humans, the excretory system consists of a pair of kidney, one pair of ureters, a urinary bladder, and a urethra. In figure 90.1, kidney are reddish brown bean-shaped structure situated between the levels of blast thoracic and third lumbar, close to the dorsal inner wall of abdominal cavity. Each kidney of an adult human measures 10 to 12 centimeter in length and 5 to 7 centimeter in width and 2 to 3 centimeter in thickness, with an average weight 120 to 170 gram. Towards the center of kidney, inner concave surface of the kidney is a notch called hilum, through which ureter, blood vessels, and nerves enter. Inner the hilum is a broader funnel-shaped space called the renal pelvis, with projection called calyxis. The outer layer of kidney is top capsule. Inside the kidney there are two zones, the outer cortex and inner medulla. The medulla is divided into few conical masses, medullary pyramids, projecting into calyces, singular calyx. The cortex extends in between the medullary pyramids as a renal column called column of Burney. Each kidney has nearly 1 million complex tubular structure called nephron, which are functional units. Each nephron has two parts, the glomerulus and the renal tubule. Glomerulus is a tuft of capillary formed by a afferent arteriole, a fine branch of renal artery. Blood from the glomerulus is carried away by afferent arteriole. The renal tubule begins with the double-walled cup-like structure called Bowman capsule, which encloses the glomerulus. Glomerulus, along with Bowman capsule, is called Malpighian body or renal capsule. The tubular continues further to form highly coiled network proximal convoluted tubule, PCT. A hairpin-shaped Henle loop is the next part of tubular, which has a descending and ascending limb. The ascending limb continues as another highly coiled tubular region called distal convoluted tubule, DCT. The DCT of many nephrons open into a straight tube called collecting duct, many of which converge and open into renal pelvis through medullary pyramid in calyces. The Malpighian corpuscle, PCT, DCT, and the nephron are situated in cortical region of kidney, whereas the loop of Henle dips into the medulla. In majority of the nephrons, the loop of Henle is short and extends only very little into the medulla. Such nephrons are called cortical nephron. In some nephrons, the loop of Henle is very long and runs dips into the medulla. These nephrons are called juxtamedullary nephron. The afferent arteriole emerging from glomerulus form a fine capillary network around the renal tubule called peritubular capillaries. A minor vessel of this network runs parallel to the Henle loop, forming U-shaped vasa recta. Vasa recta is absent in highly reduced cortical nephron. Here is 90.1 human urinary system. It is adrenal gland. This is renal artery. Here is renal vein, kidney, dorsal aorta, ureter, urinary bladder, it is urethra, and cortex, medulla, pelvis, inferior vena cava. Figure 90.2 longitudinal section diagrammatic of kidney. Medullary pyramid, renal column, calyx, renal artery, renal vein, renal pelvis, ureter, cortex, renal capsule. 9.3 diagrammatic representation of nephrons showing blood vessel, duct, and tubules. Afferent arteriole, efferent arteriole, glomerulus, Bowman capsule, and it is proximal convoluted tubule. This is distal convoluted tubule, and descending loop of Henle. This is ascending loop of Henle, and this is descending loop of Henle. This is Henle loop. 90.4 Malpighian body or renal capsule. Afferent arteriole, efferent arteriole, Bowman capsule, and this is proximal convoluted tubule. 9.2 Urine formation. Urine formation involves three main processes, namely glomerular filtration, reabsorption, and secretion, that takes place in different parts of nephron. The first step in urine formation is filtration of blood, which is carried out by glomerulus and is called glomerular filtration. On an average, 1100 to 12 ml of blood is filtered by kidney per minute, which constitute roughly 1/5 of body blood pumped out by each ventricle of the blood in a minute. The glomerular capillary blood pressure causes filtration of blood through three layers: the endothelium of glomerular blood vessel, the epithelium of Bowman capsule, and basement membrane between these two layers. The epithelial cell of Bowman capsule, called podocytes, are arranged in intricate manner so as to leave some minute spaces called filtration slit or slit pore. Blood is filtered so finely through this membrane that almost all the constituents of plasma except the protein pass on to the lumen Bowman capsule. Therefore, it is considered a process of ultrafiltration. The amount filtered found by kidney per minute is called glomerular filtration rate, GFR. GFR in healthy individual is approximately 125 per minute, that is 180 liter per day. The kidney have built-in mechanism for regulation of glomerular filtration rate. One such efficient mechanism is called out by juxtaglomerular apparatus, JGA. JGA is a special sensitive region formed by cellular modification in distal convoluted tubule and afferent arteriole at the location of their contact. A fall in GFR can activate the JG cell to release renin, which can stimulate the glomerular blood flow and thereby the GFR back to normal. A comparison of volume of filtrate found per day, 180 liter per day, with that of urine released, 1.5 liter, suggests that nearly 99% of filtrate has to be reabsorbed by the renal tubule. This process is called reabsorption. The tubular epithelial cell is in different segments of nephron perform either active or passive mechanism. For example, substances like glucose, amino acids, sodium, ion, etc. in the filter are reabsorbed actively, whereas the nitrogenous waste are absorbed by passive transport. Reabsorption of water also occurs passively in initial segment of nephron. During the urine formation, the tubular cells secrete substances like hydrogen ion, potassium and ammonium amine to filtrate. Tubular secretion is also an important step in urine formation, as it helps in the maintenance of ionic and acidic base balance of the body. 90.3 Function of the tubules. Proximal convoluted tubule, PCT. PCT is lined by simple cuboidal brush border epithelium, which increase the surface area of reabsorption. For reabsorption, nearly all the essential nutrient and 70 to 80% of electrolyte and water are reabsorbed by this segment. PCT also helps in maintaining the pH and ionic balance of the body fluid by selective secretion of hydrogen ion, ammonia and potassium ion into filtrate, and by absorption of bicarbonate ion from it. Henle loop. Reabsorption is minimum in its ascending limb. However, this region plays a significant role in maintenance of high osmolarity of medullary interstitium fluid. The descending limb of loop of Henle is permeable to water but almost impermeable to electrolyte. This concentrates the filtrate as it moves down. The ascending limb is impermeable to water but allow transport of electrolyte actively or passively. Therefore, as the concentrated filtrate pass upward, it gets diluted due to passage of electrolyte to medullary fluid. DCT, distal convoluted tubule. Conditional reabsorption of sodium ion and water takes place in this segment. DCT is also capable of reabsorption of bicarbonate ion and selective secretion of hydrogen and potassium ion and ammonia to maintain the pH and sodium potassium balance in the body. Collecting duct. This lung extends from the cortex of kidney to the inner part of medulla. Large amount of water could be reabsorbed from this region to produce a concentrated urine. This segment allows passage of a small amount of urea into medullary interstitium to keep up osmolarity. It also plays a role of maintenance of pH and ionic balance of blood by selective secretion of hydrogen ion and potassium ion. 90.5 reabsorption and secretion of major substances at different part of the nephron. Arrows indicate direction of movement of materials. Proximal convoluted tubule, distal convoluted tubule. It is cortex and this is medulla. From PCT, bicarbonate ion, sodium and salt, NaCl, water, nutrient, potassium ion released from tubules, and these H plus NH3 secrete into the tubules. Here NaCl, water and bicarbonate reabsorbed and potassium and hydrogen are secreted in the tubules. This is descending loop of Henle, and water diffuses from descending loop of Henle, and NaCl is released from ascending loop of Henle. NaCl, this is thick segment of ascending limb, this is thin segment, and NaCl urea come out from this collecting duct and get into ascending loop of Henle. This is collecting duct. Also water also come out from this collecting duct. 90.4 mechanism of concentration of urine. Mammals has the ability to produce a concentrated urine. The Henle loop and vasa recta play a significant role in this. The flow of filtrate in two limbs Henle loop is in opposite direction and thus form a counter current. The flow of blood through two limbs of vasa recta is also counter the current pattern. The proximity between the Henle loop and vasa recta as well as counter current in them help in maintaining and increasing osmolarity toward the inner medullary interstitium, that is 300 millismol in cortex to about 1200 millismol in the inner medulla. This gradient is mainly caused by NaCl and urea. NaCl is transported by ascending limb of loop of Henle and exchange with descending loop of limb of vasa recta. NaCl is returned to interstitium by the ascending portion of vasa recta. Similarly, the small amount of urea enter the thin segment of ascending limb of Henle loop, which transported back to interstitium by collecting tubule. The above described transport of substances facilitated by a special arrangement of loop of Henle and vasa recta is called counter current mechanism. This mechanism helps in maintaining concentration of gradient in medullary interstitium. Presence of such interstitial gradient help in easy passage of water from collecting duct, thereby concentrating the filtrate urine. Human kidney can produce urine nearly 4 times concentrated that initial filtrate form. 90.6 diagrammatic representation of neuron and vasa recta showing counter current mechanism. Afferent arteriole, efferent arteriole, Bowman capsule, and this is glomerulus. So this is what? This is vasa recta formed of blood vessels or capillary, and here is Bowman capsule, and we can see it is cortex and it is inner medulla and outer medulla. So एक चीज़ जो नोटिस करने वाली है भाई साहब, यहाँ देखो 300 मिलीऑस्मोल क्या है, यहाँ आप कंसंट्रेशन है, जितना ही क्या कर रहा है, जितना ही मेडुला के अंदर नीचे आ रहा है मेडुला में, उतना ही इसका क्या हो रहा है, ऑस्मोलिटी बढ़ रहा है. सबसे ज़्यादा यहाँ पे 1200 मिलीऑस्मोल, और सबसे कम कहाँ पाया जाएगा देख ले भाई, 200 मिलीऑस्मोल नियर डिस्टल कन्वोल्यूटेड ट्यूब्यूल के पास. ये देख लो, ये इम्पोर्टेंट है, याद कर सकते हो. मिनिमम कितना हो सकता है, 200 मिलीऑस्मोल. 90.5 regulation of kidney functions. The functioning of kidney is efficiently monitored and regulated by hormonal feedback mechanism involving hypothalamus, JGA, and certain extent of the heart. Osmoreceptor in the body is activated by change in blood volume, the body fluid volume, and ionic concentration. An excessive loss of fluid from the body can activate the receptor, which stimulate the hypothalamus to release antidiuretic hormone or vasopressin from the neurohypophysis. Antidiuretic facilitate the water reabsorption from latter part of tubule, thereby preventing the diuresis. An increase in body fluid volume can switch off osmoreceptor and suppress the ADS release to complete feedback. The antidiuretic hormone can also affect the kidney functioning by its constrictory effect on blood vessels. This causes an increase in blood vessel, and increase in blood pressure can increase the glomerular blood flow and thereby the GFR. The juxtaglomerular apparatus play a complex regulatory role. A fall in glomerular blood flow, glomerular blood pressure, GFR, can activate the JG cell to release renin, which convert angiotensin in blood to angiotensin first and further to angiotensin second. Angiotensin second, being powerful vasoconstrictor, increase the glomerular blood pressure and thereby GFR. Angiotensin second also activates the adrenal cortex to release aldosterone. Aldosterone causes the reabsorption of sodium and water from distal part of tubule. This also led to increase in blood pressure, GFR. This complex mechanism, generally known as renin-angiotensin mechanism. The increase in blood flow in the arteria of the heart causes the release of ANF, atrial natriuretic factor. ANF can cause vasodilation, dilation of blood vessel, and thereby decrease the blood pressure. ANF mechanism therefore act as check on renin-angiotensin mechanism. 90.6 maturation. Urine found by the nephrons ultimately carried out by urinary bladder when it's stored televoluntary signal and given by nervous system CNS. This signal is initiated by stretching of urinary bladder and it gets filled with urine. In response to stress receptor on the body wall, the bladder sends signal to CNS. The control nervous system passes the motor message to stimulate the contraction of smooth muscle of bladder and simultaneously relaxation of urethra sphincter, causing the release of urine. The process of release of urine called micturition, and the neural mechanism causing it called micturition reflex. An adult human excretes on an average 1 to 1.5 liters of urine per day. The urine formed in a light yellow colored watery fluid which is slightly acidic, pH 6, and has characteristic odor. On an average, 25 to 30 grams of urea is excreted out per day. Various conditions can affect the characteristic of urine. Analysis of urine helps in clinical diagnosis many metabolic disorders as well as malfunctioning of kidney. For example, presence of glucose, glycosuria, and ketone body, ketonuria in urine indicative of diabetes mellitus. 90.7 Role of other organs in excretion. Other than kidney, lungs, liver, skin also help in elimination of excretory waste. Our lung remove large amount of carbon dioxide, approximately 200 ml per minute, and also significant quantity of water every day. Liver, the largest gland in our bodies, secrete bile-containing substances like bilirubin, biliverdin, cholesterol, degraded steroidal hormones, vitamins, and drugs. Most of these substances ultimately pass out along with digestive wastes. The sweat and sebaceous gland in the skin can eliminate certain substances through their secretion. Sweat produced by sweat gland is watery fluid containing NaCl, a small amount of urea, lactic acid, etc. Though the primary function of sweat is to facilitate a cooling effect on the body surface, it also helps in removal of some of the wastes mentioned above. Sebaceous gland eliminates certain substances like sterol, hydrocarbon, and waxes through sebum. This secretion provides protective oil covering for skin. Do you know that a small amount of nitrogenous waste could eliminate through saliva too? 90% disorder of excretory system. Malfunctioning of kidney can lead to accumulation of urea in blood and condition called uremia, which is highly harmful and may lead to kidney failure. In such patients, the urea can be removed by a process called hemodialysis. During the process of hemodialysis, the blood drained from convenience artery is pumped into dialyzing unit called artificial kidney. Blood drained from the convenience artery is pumped into dialyzing unit after adding anticoagulant like heparin. The unit contains the coiled cellophane tube surrounded by fluid, dialyzing fluid having the same composition as that of plasma except nitrogenous waste. The porous cellophane membrane of a tube allows the passage of molecules based on concentration gradient. As nitrogenous waste are absent in the dialyzing fluid, these substances freely move out, thereby clearing the blood. The cleared blood flow pumped back the body through a vein after adding antiheparin to it. This method is a boom for thousands of uremic patients all over the world. Kidney transplantation is ultimate method in the correction of acute renal failure, kidney failure. A functioning of kidney is used in transportation from a donor, preferably a close relative, to minimize its chance of rejection of immune system to the host. Modern clinical procedures have increased success rate of such complicated technique. Renal calculi. Stone or insoluble mass of crystallized salt, oxalates, etc. form within kidney. Glomerulonephritis. Inflammation of glomerular kidney. Summary. Many nitrogen containing substances, iron, carbon dioxide, water, etc. that accumulate in the body have to be eliminated. Nature of nitrogenous waste formed and their excretion vary among animals, mainly depending on habitat, availability of water. Ammonia, urea, and uric acid are the major nitrogenous waste excreted. Proton, nephridium, malphizian tubules, green gland, and kidney are the common excretory organ in animals. They not only eliminate nitrogenous waste but also help in maintenance of ionic acid base balance in body fluid. In humans, the excretory system consists of one pair of kidney, a pair of ureters, a urinary bladder, and a urethra. Each kidney has over millions of tubular structures called nephrons. Nephron is a functional unit of kidney and has two portions, glomerulus and renal tubule. Glomerulus is a tuft of capillary formed from afferent arterioles, fine branches of renal artery. Renal tubule starts with a double-walled Bowman capsule and is further differentiated into proximal convoluted tubule (PCT), Henle loop (HCl), and distal convoluted tubule (DCT). The DCT of many nephrons join to a common collecting duct, many of which ultimately open into the renal pelvis through medullary pyramid. The Bowman capsule encloses the glomerulus to form malphizian or renal capsule. Urine formation involves three main processes: filtration, reabsorption, and secretion. Filtration is a non-selective process performed by glomerulus using glomerular capillary blood flow pressure. About 1200 ml of blood is filtered by glomerulus per minute to form 120 ml of filtrate in Bowman capsule per minute. JJ is specialized portion of nephron play a significant role in regulation of GFR. Nearly 99% of reabsorption of filtrate takes place through different part of nephron. Proximal convoluted tubule is the major site of reabsorption and selectively secretion. Henle loop primarily help to maintain osmolar gradient 300 milliosmol to 1200 milliosmol within the kidney interstitium. Distal convoluted tubule and collecting duct allow extensive reabsorption of water and certain electrolytes, which help in osmoregulation. Ammonia could be secreted into filtrate by tubules to maintain ionic balance and pH of body fluid. A counter-current mechanism operates between the two limbs of blood of the loop of Henle and those vascular capillary parallel to Henle loop. The filtrate gets concentrated as it moves down the descending limb but diluted by ascending limb. Electrolyte and urea are retained in interstitium, by which arrangement DCT collecting duct concentrates the filtrate about 4 times, from 300 to millismol to 1200 millismol. An excellent mechanism of conservation of water. Urine is stored in urinary bladder till the voluntary signal from central nervous system carries out its release through urethra micturition. Skin, lungs, liver also assist in excretion. Chapter completed. So by chapter 20, locomotion and movement. ये चैप्टर स्टार्ट करने के लिए करेंगे 4-5 से, तो 3 मिनट का रेस्ट। मिलते हैं भाई छोटा सा नमूना ब्रेक के बाद ब्रो। अच्छा चलता हूँ दुआओं में याद रखना, मेरे जिक्र का सुबह पे स्वाद रखना, मेरे आते हैं ब्रो, 3 मिनट बाद मिलते हैं बड़े भाई लोग से। सारी दुनिया से जीत के मैं आया हूँ इधर, तेरे आगे ही तो वेलकम बैक भाई लोग, टाइम हो गया। वेलकम बैक मतलब लोकोमोशन एंड मोमेंट। तो इसमें क्या पढ़ेंगे, 20.1 टाइप ऑफ मोमेंट, 20.2 मसल्स, 20.3 स्केलेटल मसल, 20.4 जॉइंट्स, 20.5 डिसऑर्डर्स ऑफ मस्कुलर एंड स्केलेटल सिस्टम। Movement is the one of significant feature of living being. Animals and plant exhibit wide range of movement. Streaming of protoplasm is in unicellular organism like amoeba is a simple form of movement. Movement of cilia, flagella and tentacles are shown by many organisms. Human being can move limbs, jaw, eyelids, tongue, etc. Some of the movement result in change in places and location. Such voluntary movement is called locomotion. Walking, running, climbing, flying, swimming, all some forms of locomotory movements. Locomotory structure need not be different from those affecting other type of movement. For example, paramecium cilia help in movement of food through cytopharynx and locomotion, as well as in hydra. Hydra can use its tentacles for capturing its prey and also use them for locomotion. We use limbs for change in body postures and locomotion as well. The above observation suggests that movement and locomotion cannot be studied separately. The two may be linked by stating that all locomotion are movements, but all movements are not locomotion. Methods of locomotion performed by animals vary with their habitats and demand of situations. However, locomotion is generally for Search of food, shelter, mate, suitable breeding grounds, favorable climatic condition, or to escape from enemies or predators. 20.1 Type of movement. Cells of human body exhibit three types of movement, namely amoeboid, ciliary, or muscular. Some specialized cells in our body, like macrophages, leukocytes in blood, exhibit amoeboid movement. It is affected by pseudopodia found by streaming protoplasm, as in amoeba. Cytoskeleton elements like microfilament are also involved in amoeboid movement. Ciliary movement occur in some of our internal tubular organs, which are lined by ciliated epithelium. The coordinated movement of cilia in the trachea help us in removing dust particles and some of the foreign substances inhaled along with atmospheric air. Passage of ova through the female reproductive tract is also facilitated by ciliary movement. Movement of our limbs, jaw, tongues, etc. require muscular movement. The contractile property of muscle effectively use the locomotion and other movement by human beings and majority of multicellular organisms. Locomotion requires perfect coordinated activity of muscular, skeletal, and neural system. In this chapter, you will learn about the type of muscles, their structure, mechanism, and contraction, and important aspect of skeletal muscle. 20.2 Muscle. You have studied in chapter 8 that cilia and flagella are the outgrowth of cell membrane. Flagellar movement help in the swimming of spermatozoa, maintenance of water current in the canal system of sponges, and locomotion of protozoans like Euglena. Muscles is a specialized tissue of mesodermal origin. About 40 to 50% of body weight of human adult is contributed by muscles. They have a special property like excitability, contractility, extensibility, and elasticity. Muscle have been classified using different criteria, namely location, appearance, and nature of regulation of their activities. Based on their location, three types of muscle are identified: skeletal, visceral, and cardiac. Skeletal muscle are closely associated with skeletal component of the body. They have a stripped up appearance under microscope and hence are called striated muscle. As their activities are under voluntary control of nervous system, as they are known as voluntary muscle too. They are primarily involved in locomotory action and change in body posture. Visceral muscle are located in the inner walls of hollow visceral muscles of the body, like alimentary canal, reproductive tract, etc. They do not exhibit any striation and are smooth in appearance. Hence, they are called smooth muscle, non-striated muscle. Their activities are not under the voluntary control of nervous system and therefore known as voluntary muscle. They assist, for example, in the transportation of food through digestive tract and gametes through genital tract. As the name suggests, cardiac muscle are the muscle of heart. Many cardiac muscle cells assemble in branching pattern to form a cardiac muscle. Based on appearance, cardiac muscle are striated. They are involuntary in nature as the nervous system does not control their activities directly. Let us examine a skeletal muscle in detail and understand structure and mechanism of contraction. Each organized skeletal muscle in our body is made up of number of muscle bundle fascicles held together by common collagenous connective tissue called fascia. Each muscle bundle contains number of muscle fibers. Each muscle fiber is lined by a plasma membrane called sarcolemma, enclosing the sarcoplasm. Muscle fiber is syncytium, as the sarcoplasm contains many nuclei. The endoplasmic reticulum, that is sarcoplasmic reticulum of muscle fiber, is the storehouse of calcium ion. A characteristic feature of muscle fiber is the presence of large number of parallelly arranged filaments in the sarcoplasm called myofilament or myofibrils. Each myofibril has alternate dark and light band on it. A detailed study of myofibril has established that striated appearance is due to distribution pattern of two important proteins, actin and myosin. The light band contain actin and is called I band or isotropic band, whereas the dark band contain called A or N isotropic band contain myosin. Both the protein are arranged in rod-like structure parallel to each other and also longitudinal axis of myofibril. Actin filament are thinner as compared to the myosin filament, hence are commonly called thin and thick filament respectively. In the center of each I band is an elastic fiber called Z line which bisect it. The thin filament are firmly attached to Z line. The thick filament in the A band are also held together in the middle of this band by a thin fibrous membrane called M line. The A and I band are arranged alternatively throughout the length of myofibril. The portion of myofibril between two successive Z line is considered as functional unit of contraction and is called sarcomere. In the resting state, the edge of thin filament is either side of the thick filament partially overlap the free end of thick filament, leaving the central part of thick filaments. This central part of thick filament not overlapped by thin filament is called H zone. Figure 20.2 diagrammatic cross section view of muscle fiber showing the muscle bundle and muscle fibers. This is fascicle muscle bundle. This is muscle fiber or muscle cell, and this is sarcolemma, and this is blood capillary. Diagram 12.20.2 diagrammatic representation of anatomy of muscle fiber and a sarcomere. This is Z line. This is also Z line, and these are I band. This gap is known as I band, and this is A band. थिक मायोसिन जो है जहाँ तक, और ये गैप है इसको एच जोन बोलते हैं, और ये यहाँ से यहाँ तक क्या दो जेड लाइन के बीच का गैप सार्कोमियर. 20.2.1 structure of contractile protein. Each actin thin filament is made up of two filamentous actin helically on to each other. Each of actin is polymer of monomeric G globular actins. Two filaments of another proteins, tropomyosin, also run close to F actins throughout its length. A complex protein troponin is distributed at regular interval on tropomyosin. In the resting state, a subunit of troponin marks the active binding site for myosin on the active filament. Each myosin thick filament is also a polymerized protein. Many monomeric protein called meromyosin constitute one thick filament. Each meromyosin has two important parts, a globular head with a short arm and a tail. The former being called the heavy meromyosin HMM and the latter the light meromyosin. The heavy meromyosin component, that is the head and the short arm, project outward at regular distances and angle from each other from the surface of polymerized myosin. Filament and is also known as cross arm. The globular head is an active ATPase enzyme and has binding site for ATP and active site for actin. An actin filament, thin filament, and myosin, meromyosin. It is troponin, tropomyosin, and there are two F actin. And this is myosin, cross arm, head एंड एटीपी बाइंडिंग साइट, ये जो नीचे है एटीपी बाइंडिंग साइट है, और एक्टिन बाइंडिंग साइट का है ऊपर है. Muscle contraction is initiated by signals sent by central nervous system CNS by the motor neuron. A motor neuron along with the muscle fiber connected to constitute motor fiber. The junction between a motor neuron and sarcolemma of muscle fiber is also called a neuromuscular junction or motor end plate. A neural signal reaching this junction releases neurotransmitter acetylcholine, which generates an action potential in the sarcolemma. This is spread through the muscle fiber and causes the release of calcium ion into the sarcoplasm. Increase in calcium ion level led to binding of calcium with a subunit troponin on actin filament and thereby remove the masking of active site for myosin. Utilizing the energy from ATP hydrolysis, the myosin head now bound to expose active site on actin to form cross bridge. This pull the attached actin filament towards center of A band. The jet line attached to these actin are also pulled inward, thereby causing a shortening of sarcomere, that is contraction. It is clear from the above step that during shortening of muscle, that is contraction, the I band get reduced where the A band retain the length. The myosin releasing the ADP and phosphate 1 goes back to relaxed state. A new ATP bind to a cross bridge and broken. The ATP again hydrolyzed by myosin head and the cycle of cross bridge formation and breakage are repeated, causing further sliding. The process continues till calcium ion pumped back to the sarcoplasmic system, resulting in the masking of actin protein filaments. The cause of return to Z line back to their original position, that is relaxation. The reaction time of fibers can vary different muscles. Repeated activation of muscle fiber can lead to the accumulation of lactic acid due to anaerobic breakdown of glycogen in them, causing fatigue. Muscle contain a red colored oxygen storing pigment called myoglobin. Myoglobin content is high in some of the muscles, which give a reddish appearance. Such muscles are called the red fibers. These muscles also contain plenty of mitochondria, which can utilize a large amount of oxygen stored in them for ATP production. These muscles thereby can also be called aerobic muscle. On the other hand, some muscles possess very less quantity of myoglobin and therefore appear pale or whitish. These are white fibers. Number of mitochondria are also few in them, but the amount of sarcoplasmic reticulum is high. They depend on anaerobic process for energy. 20.5 Sliding filament theory for muscle contraction, moment of thin filament and relative size of I band and H zone. This is Z line, and this gap is known as H zone. This is myosin and it is also myosin, and this is actin fiber, and this space between two myosin is I band, and this total यहाँ से पूरे मायोसिन का जो लेंथ है इसको ए बैंड बोलेंगे. ठीक, ये रिलैक्स स्टेट है. कंट्रैक्ट में क्या हुआ, छोटा हो रहा है इधर, तो ये क्या, ये जो एच जोन भी कम होगा और जेड लाइन के बीच का भी दूरी कम होगा कंट्रैक्शन में. मैक्सिमम कंट्रैक्ट जब हो गया, यहाँ पे देखो क्या है, ए बैंड कांस्टेंट है यहाँ पे. 20.3 Skeletal system. Skeletal system consists of framework of bones and a few cartilages. This system has significant role in movement shown by body. Imagine chewing food without jaw bones and walking around without limbs bone. Bone is cartilage are specialized connective tissue. The former has a very hard matrix due to calcium salt, and and the latter has slightly pliable matrix due to chondrian salt. In human beings, the system made of 206 bones and few cartilage. It is grouped into two principal divisions: the axial and appendicular skeleton. Axial skeleton comprises of 80 bones distributed along with main axis of the body. The skull, vertebral column, sternum, and ribs constitute axial skeleton. The skull is composed of two sets of bones, the cranial and facial, that total 22 bones. Cranial bones are 8 in number. They form the hard protective outer covering, cranium for the brain. The facial region is made of 14 skeletal elements which form front part of skull. A single U-shaped bone called hyoid is present in base of buccal cavity, and it is also included in the skull. Each middle ear contains 3 tiny bones, malleus, incus, and stapes, collectively called ear ossicles. The skull region articulates with superior region of vertebral column with the help of 2 occipital condyles, dicondylic skull. ये क्या है फ्रंटल बोन, ये क्या है parietal, ये क्या है टेम्पोरल बोन, ये क्या है occipital बोन, ये क्या है occipital condyle, this is sphenoid bone, this is ethmoid bone, ये क्या है lacrimal, ये क्या है nasal bone, this is zygomatic bone, this is maxilla, and this is mandible, and this is hyoid bone. Diagrammatic view of human skull. Our vertebral column is formed by 26 serially arranged units called vertebrae and is dorsally placed. It extends from the base of skull and constitutes the main framework of trunk. Each vertebrae has a central hollow portion neural canal through which the spinal cord passes. First vertebrae is the atlas and it articulates with the occipital condyles. The vertebral column is differentiated into cervical C7, cervical thoracic 12, lumbar 5, sacral 1 fused, and coaxial 1 fused. Region starting from the skull. The number of cervical vertebrae are 7 in almost all mammals, including human beings. The vertebral column protects the spinal cord, supports the head, and serves as the point of attachment for the ribs and musculature of the back. Sternum is a flat bone at the ventral midline of thorax. Cervical vertebrae, thoracic vertebrae, lumbar, sacral fused, coaxial, intervertebral disc, vertebral column of right lateral view. There are 12 pair of ribs. Each rib is a thin flat bone connected dorsally to vertebral column and ventral to sternum. It has two articulation surface on its dorsal end and is hence called bicephalic. First 7 pair of ribs is called true ribs. Dorsally they are attached to thoracic vertebrae and ventrally connected to sternum with the help of hyaline cartilage. The 8th, 9th and 10th pairs of ribs do not articulate directly with the sternum but join the 7th ribs with the help of hyaline cartilage. These are called vertebrochondral or false ribs. Last two pair, 11th and 12th of ribs are not connected ventrally and are therefore called floating ribs. Thoracic vertebrae ribs and sternum together form rib cage. The bones of limbs along with their girdles constitute appendicular skeleton, each limb made up of 30 bones. The bones of hand, forelimb and humerus, radius, ulna, carpus, wrist bones 8 in number, metacarpus, palm bone 5 in number, phalanges digit 14 in numbers, femur, thigh bone, the longest bone, tibia and fibula, femur, thigh bone longest and tibia, fibula, tarsal ankle, there are 7 bones, metatarsal 5 in numbers, phalanges 14 in numbers, are bones of legs, hind limbs. A cup shaped called patella cover the knee ventrally kneecap. 20.8 ribs and rib cage. This is sternum, ribs, vertebral column 1 to 7 true ribs, 8, 9, 10 false rib, 11 and 12 floating ribs. Right arm pectoral girdle arms. This is clavicle bone. This is scapula, humerus, radius, ulna, carpals, metacarpals, phalanges. Pectoral and pelvic bones help in the articulation of upper and lower limbs respectively with the help of axial skeleton. Each girdle is formed of two halves. Each half of the pectoral girdle consists of clavicle and scapula. Scapula is a large triangular flat bone situated in dorsal part of thorax between 2nd and 7th ribs. The dorsal flat triangular body scapula has slightly elevated ridge called spine which projects as flat expanded process called acromion. The clavicle articulate with this. Below the acromion is depression called glenoid cavity which articulate with the head and humerus to form shoulder joint. Each clavicle is long slender bone with the two curvature. This bone is commonly called collarbone. Pelvic girdle consists of two coaxial bones. Each coaxial bone is formed by the fusion of three bones, ilium, ischium, and pubis. At the point of fusion of two bones is cavity called acetabulum, to which thigh bone articulate. The two bones of pelvic girdle meet ventrally to form pubic symphysis containing fibrous bone. 20.10 Pelvic girdle and lower limbs bones frontal. This is ilium, this is coaxial bone, sacrum, this is pubis, ischium, femur, patella, tibia, this is fibula, this is tibia and this is fibula. मोटा जो है उसका है tibia और fibula. Tarsals, metatarsals and phalanges. 25.4 Joints. Joints are essential for all types of movement involving bony parts of the body. Locomotive movement are no exception to this. Joints are point of contact between bones or between bones and the cartilages. Forces generated by muscles is used to carry out movement through joints, where the joints act as fulcrum. The movability at these joints vary depending on different factors. Joints have been classified into three major structural forms, namely fibrous, cartilaginous, and synovial. Fibrous joints do not allow any movement. This type of joint is shown by flat skull bones which fuse end to end with the help of dense fibrous connective tissue in the form of sutures to form cranium. In cartilaginous joint, the bone involved in joint involved together with the help of cartilages. The joint between the adjacent vertebrae in the vertebral column is of this pattern and is permit limited movement. Synovial joint are characterized by the presence of fluid-filled synovial cavity between articulating surface and joint two bones. Such an arrangement allows considerable movement. These joints help in locomotion and mainly the other movements. Ball and socket joints between humerus and pectoral girdle, hinge joint between knee joint, pivoted joint between atlas and axis, gliding joint between carpals and saddle joint between carpals and metacarpals and thumb, and some example. 20.5 Disorders of muscle and skeletal system. Myasthenia gravis, autoimmune disorder affecting neuromuscular junction leading to the fatigue, weakening and paralyzing skeletal muscle. Muscular dystrophy, progressive degeneration of skeletal muscle mostly due to genetic disorder. Tetany, rapid response wild contraction in muscle due to low calcium ion in body fluid. Arthritis, inflammation of joints. Osteoporosis, age-related disorder characterized by decrease in bone mass and increase in chance of fracture. Decreased level of estrogen is common cause. Gout, inflammation of joint due to accumulation of uric acid. Summary: movement is an essential feature of all living beings. Protoplasmic streaming, ciliary movement, movement of fins, limb, wings, etc. are some forms exhibited by animals. A voluntary movement which causes the animal to change its place is called locomotion. Animals move generally in search of food, shelter, mate, breeding ground, better climate, and protect themselves. The cell of human body exhibits a wide ciliary and muscular movement. Locomotion of many of other movements require coordinated muscular activities. Three types of muscles present in our body. Skeletal muscles are attached to skeletal element. They appear in striated and voluntary in nature. Visceral muscle present in inner wall of visceral organs and non-striated and involuntary. Cardiac muscle are the muscles of heart. They are striated, branched and involuntary. Muscle possess excitability, contractibility and extensibility and elasticity. Muscle fiber is anatomically unit of muscle. Each muscle fiber has many parallelly arranged myofibrils. Each myofibril contains many serially arranged units called sarcomere, which are functional units. Each sarcomere has a band made of thick myosin filaments and two half-eye bands made up of thin actin filament on either side, marked by Z lines. Actin and myosin are polymerized protein with contractility. The active site of myosin on resting actin filament are marked by protein troponin. Myosin head contains ATPase and has ATP binding site and active site for actin. A motor neuron carries signal for muscle fiber which generate an action potential in it. This causes the release of calcium ion from sarcoplasmic reticulum. Calcium ion activates actin which bind to myosin head to form cross bridge. These cross bridge pull out the actin filament, causing them to slide over the myosin filament and thereby causing contraction. Calcium ion are then returned back to sarcoplasmic reticulum which inactivate the actin. Cross bridges are broken and the muscle relax. Repeated stimulation of muscle led to fatigue. Muscle are classified as red-white fibers based on primarily on amount of red-colored myoglobin pigment in them. Bones and cartilage constitute our skeletal system. The skeletal system is divisible into axial and appendicular. Skull, vertebral column, and ribs and sternum constitute the axial skeleton. Limbs, bone, and girdles form appendicular skeletal. Three types of joints are formed between bones and cartilage fibers: cartilaginous and synovial. Synovial joints allow considerable movement and therefore play a significant role in locomotion. So by 4:33 to 4:35 तक रेस्ट और ब्रेक, उसका अपना चलेगा क्या, न्यूरल कंट्रोल एंड कोऑर्डिनेशन, चैप्टर 21. तब तक के लिए ओके, बाय. लेट्स स्टार्ट ब्रो, टाइम हो गया यार. (20.1) Neural system. 20.2 human neural system. 21.3 neuron as structure and functional unit of neural system. 20.1 central neural system. 21.5 reflex action and reflex are. 21.6 sensory reception and processing. As you know, the function of organs and organ system in our body must be coordinated, maintaining homeostasis. Coordination is the process through which two or more organs interact and complement the functioning of one another. For example, when we do physical exercises, the energy demand is increased for maintaining an increase in muscular activity. The supply of oxygen is also increased. The increased supply of oxygen necessitates an increase in rate of respiration, heartbeat, and increase in blood flow by the way blood vessels. When physical exercise is stopped, the activities of nerves, lungs, heart, and kidney gradually return to their normal condition. Thus, the functions of muscle, lung, heart, blood vessel, kidney, and other organs are coordinated while performing physical exercises. In our body, the neural system and endocrine system jointly coordinated and integrated all activities of organs so that they function as in a synchronized fashion. The neural system provides an organized network of point-to-point connection for quick coordination. The endocrine system provides chemical integration through hormone. In this chapter, you will learn about the neural system of human, mechanism of neural coordination like transmission of nerve impulses, impulse conduction across synapses and physiology of reflex action. Neural system. Neural system of all animals is composed of highly specialized cells called neurons, which can detect, receive, and transmit different kind of stimuli. The neural organization is very simple in lower invertebrates. For example, in hydra, it is composed of network of neurons. The neural system is better organized in insect, where the brain is present along with number of ganglia and neural tissue. The vertebrates have more developed neural system, 21.2 human neural system. The human neural system is divided into two parts. First, the central neural system CNS, and second, the peripheral nervous system PNS. The CNS include brain and spinal cord and is the site for information processing and control. The peripheral nervous system of all type of nerves, associated type, control nervous system, and the nerve fiber PNS, two type, afferent fibers and efferent fibers. Afferent nerve fiber transmit impulse from tissue or organ to control nervous system, and afferent transmit regulatory impulse from the control nervous system and concerned peripheral tissue organs. The peripheral nervous system divided into two divisions called somatic neural system and autonomic neural system. The somatic neural system relays impulses from central nervous system to skeletal muscle, while the autonomic nervous system transmits impulses from central nervous system to voluntary organ and smooth muscle of the body. The autonomic neural system is further classified into sympathetic neural system and parasympathetic neural system. Visual nervous system is the part of peripheral nervous system that comprises the whole complex of nerves, fibers, ganglion, and plexus, by which impulses travel from central nervous system to viscera and from viscera to central nervous system. 20.1 neuron is structural and functional unit of neural system. A neuron is microscopic structure composed of three major parts, namely cell body, dendrite, and axon. This cell body consists of cytoplasm with typical cell organelles and certain granular bodies called nuclear granules. Short fiber which branch rapidly and project out of cell body also contain nuclear granules and called dendrites. These fiber transmit impulse towards the cell body. The axon, long fiber and distal end of which each branch terminates a bulb-like structure called synaptic knob, which possess synaptic vesicles containing chemical and called neurotransmitter. The axon transmit nerve impulses away from the cell body to synapses or to neuromuscular junction. Based on the number of axon and dendrite, the neuron are divided into three types: multipolar, with one axon and two or more dendrites, found in cerebral cortex; bipolar, with one axon and one dendrite, found in retina of eyes; and unipolar, cell body with one axon only, found usually in embryonic stage. There are two types of axon, namely myelinated and non-myelinated. The myelinated nerve fibers are enveloped with Schwann cells, which form a myelin sheath around the axon. The gaps between two adjacent myelin sheaths are called nodes of Ranvier. Myelinated nerve fibers are found in spinal and cranial nerves. Myelinated nerve fibers is included by a Schwann cell that does not form myelin sheath around axon and is commonly found in autonomous and somatic neural system. Structure of neuron. These are dendrites, nuclear granules, cell body. This is nucleus. This is Schwann cell. This is axon. This is myelin sheath. This is nodes of Ranvier. This is axon terminal. This is synaptic knob. 21.3.1 Generation and conduction of nerve impulses. Neurons are excitable cells because their membranes are polarized state. Do you know why the membrane of neuron is polarized? Different types of ion channels are present on the neural membrane. These ion channels are selectively permeable to different ions. When a neuron is not conducting any impulse that is resting, the axonal membrane is comparatively more permeable to potassium ion K+, and nearly impermeable to sodium ion Na+. Similarly, the membrane is impermeable to negatively charged protein present in the exoplasm. Consequently, the exoplasm inside exon contain high concentration of potassium ion and negative charge protein to low concentration of sodium ion. In contrast, the fluid outside exon contain a low concentration of potassium ion or high concentration of sodium ion and thus form concentration gradient. These ionic gradient across the resting membrane are maintained by active transport of ion by sodium-potassium pump, which transport 3 sodium outside and 2 potassium into the cell. As a result, outer surface of the axonal membrane possesses positive charge while the inner become negatively charged, therefore polarized. The electrical potential difference across the resting plasma membrane is called resting potential. You might be curious to know that the mechanism of generation of nerve impulse and conduction along the an axon. When a stimulus is applied to the site that is at a point A on polarized membrane, the membrane at the site A become freely permeable to sodium ion. This led to the rapid influx of sodium ion followed by reversal of polarity at that site. That is the outer surface of membrane become negatively charged and the inner side become positively charged. The polarity of membrane at site A is thus reversed and hence depolarized. The electric potential difference across the plasma membrane at the site A is called the action potential, which is in fact termed as nerve impulse. At the site immediately ahead the axon, that is the site B membrane, has positive charge on the outer surface and negative charge on inner surface. As a result, the current flow on inner surface from site A to site B. On the outer side surface, current flow from site B to site A. The complete circuit flow, current flow. Hence, the polarity of at the site is reversed and action potential is generated at site B. Thus, the impulse action potential generated at site A arrives at site B. The sequence of repeated along with length of axon and consequently the impulse conducted. The rise of stimulus in induced permeability of sodium extremely short-lived. It is quickly formed by rise in permeability of potassium ion within the fraction of second. Potassium diffuses outside membrane and restores the resting potential of membrane at site excitation, and fibers become one more responsive to further stimulation. Figure 21.2 diagrammatic representation of impulse conduction through axonal membrane. Transmission of impulse. A nerve impulse is transmitted from one neuron to another neuron through junction called synapses. A synapsis is formed by membrane of a presynaptic neuron and postsynaptic neuron, which may or may not be separated by gap called synaptic cleft. There are two types of synapses, namely electrical synapses and chemical synapses. At the electrical synapses, the membrane pre and postsynaptic neuron are in very close proximity. Electrical current can flow directly from one neuron into the other across these synapses. Transmission of an impulse across electrical synapses is very similar to impulse conduction along a single axon. Impulse transmission across electrical synapses is always faster than that of across chemical synapses. Electrical synapses is rare in our body. At the chemical synapses, membranes of pre and post synaptic neurons are separated by a fluid filled space called synaptic cleft. Do you know how the presynaptic neuron transmit an impulse action potential across the synaptic cleft to the postsynaptic neuron? Chemicals called neurotransmitters are involved in the transmission of impulse at these synapses. The axon terminals contain vesicles filled with these neurotransmitters. When an impulse action potential arrives at the axon terminal, it stimulates the movement of the synaptic vesicles toward the membrane where they fuse with the plasma. Plasma membrane release their neurotransmitter in the synaptic cleft. The release of neurotransmitter binds to a specific receptor present on the postsynaptic membrane. This binding opens ion channel, allowing the entry of ion generate new potential in postsynaptic neuron. The new potential developed may be either excitatory or inhibitory. Here diagram showing axon terminal and of synapses. This is axon terminal, synaptic vesicle, presynaptic membrane, synaptic cleft, and this is postsynaptic membrane, and this is synapses, this is receptors, and here are neurotransmitters. 21.4 Central nervous system. The brain is the central information processing organ of our body and acts as command and control system. It controls the voluntary movement of and balance of the body, functioning vital involuntary organs, that is lung, kidney, air, heart and kidney. Thermoregulation, hunger and thirst, circadian 24-hour rhythms of our body, activities of several endocrine glands and human behaviors. It is also the site of processing of vision, hearing, speech, memory, intelligence, emotion and thought. The human brain is well protected by skull. Inside the skull, the brain is covered by cranial meninges consisting of outer layer called dura mater and a very thin layer, middle layer called arachnoid, and the inner layer which is contact with brain tissue called pia mater. The brain can be divided into three major parts: first forebrain, second midbrain, and third is hindbrain. Diagram showing sagittal. This is sagittal section of human brain. Cerebral hemisphere, corpus callosum, and this is cerebral aqueduct. This is cerebrum contain forebrain contains cerebrum, thalamus and hypothalamus. This is midbrain, and hindbrain contain pons, cerebellum, medulla, and this is a spinal cord. 21.4.1 Forebrain. The forebrain consists of cerebrum, thalamus and hypothalamus. Cerebrum forms major part of human brain. A deep cleft divides cerebrum longitudinally into two halves, which are termed as left and right cerebral hemisphere. The hemispheres are connected by tract of nerve fibers called corpus callosum. The layer of cells which cover the cerebral hemisphere called cerebral cortex and is thrown into prominent fold. The cerebral cortex is referred to as grey matter due to greyish appearance. The neuron cell bodies are concentrated here, giving the color. The cerebral cortex contains motor areas, sensory areas, and large regions that are neither clearly sensory nor motor in function. These regions are called association areas, are responsible for complex functions like intersensory association, memory, and communication. Fibers of the tract are covered with the myelin sheath, which constitute the inner part of cerebral hemisphere. They give an opaque white appearance of layer, hence is called white matter. This cerebrum wraps around a structure called thalamus, which is a major coordinating center of sensory and motor signaling. Another very important part of brain is called hypothalamus, lies at the base of thalamus. The hypothalamus contains number of centers which control body temperature, urge of eating, drinking, and it also contains several group of neurosecretory cells which secrete hormones called hypothalamic hormone. The inner part of cerebral hemisphere and a group associated deep structure called amygdala, hippocampus, etc. form a complex structure called limbic lobe or limbic system. Along with hypothalamus, it is involved in regulation of sexual behavior, expression, emotion, and reaction. That is excitement, pleasure, and rage, and fear, and motivation. 21.4.2 Midbrain. The midbrain is located between the thalamus, hypothalamus of the forebrain and pons of hindbrain. A canal called cerebral aqueduct passes through midbrain. The dorsal portion of midbrain consists mainly of four round swelling lobes called corpora quadrigemina. 21.4.3 Hindbrain. The hindbrain comprises of pons, cerebellum, medulla, also called the medulla oblongata. Pons consists of fibrous tract that interconnect different regions of brain. Cerebellum has a very convoluted surface in order to provide additional space for many more neurons. The medulla of the brain is connected to spinal cord. The medulla contains center which control respiration, cardiovascular reflexes, and gastric secretion. Three major regions make up the brain: stem, midbrain, and pons and medulla oblongata. Brain stem form connection between brain and spinal cord. सो भाई कंप्लीट हो गया, यहाँ तक था. अब समरी देख लेते हैं इसका. न्यूरल सिस्टम कोऑर्डिनेट द इंटीग्रेशन. ये आइज का है, ये हट गया. ईयर का ये नहीं पढ़ना है. The neural system coordinate integrate function as well as metabolic homeostatic activities of all organs. Neurons, the functional unit of neural system, are excitable cells due to differential concentration gradient of ions across membrane. The electrical potential difference across the resting neural membrane is called resting potential. The nerve impulses is conducted along the axon membrane in the form of wave depolarization and repolarization. A synapse is formed by membranes of presynaptic neuron and postsynaptic neuron, which may or may not be separated by a gap called synaptic cleft. Chemical environment transmission of impulses at chemical synapses are called neurotransmitters. Human system consists of two parts: central nervous system and peripheral nervous system. The central nervous system consists of brain and spinal cord. The brain can be divided into three major parts: first forebrain, second midbrain, and third hindbrain. The forebrain consists of cerebrum, thalamus, and hypothalamus. The cerebrum is longitudinally divided into two halves connected by corpus callosum. A very important forebrain called hypothalamus control body temperature, eating, drinking, and inner part of cerebral hemisphere group associated deep structure form a complex structure called limbic system, which is connected with olfaction, autonomic responses, regulation of sexual behavior, expression of emotion, reaction, and motivation. The midbrain receive and integrate visual, tactile, and auditory impulse input. The hindbrain comprises of pons, cerebellum, and medulla. The cerebellum integrates information received from semicircular canal of ear and auditory system. The medulla contains center of control, respiration, cardiovascular reflex, and gastric secretion. Pons consist of fiber tracts that interconnect different regions of brain. The entire process of involuntary response to peripheral nervous stimulation is called reflex action. Information regarding change in environment received by control nervous system through sensory organ which proceed and analyzed. Signals are then sent to necessary adjustment. The wall of human eyeball are composed of ये हटाओ NCERT में हटी है. चलो भाई 4:55 से 5 PM तक रेस्ट. Then we start chemical coordination and integration. टाइम हो गया मेरा भाई, चलो स्टार्ट करते हैं। चैप्टर 22 केमिकल कोऑर्डिनेशन एंड इंटीग्रेशन। 22.1 endocrine glands and hormones। 22.2 human endocrine system। 22.3 hormone of heart, kidney and gastrointestinal tract। and 22.2 mechanism of hormone. You have already learned that neural system provides a point-to-point rapid coordination among organs. The neural coordination is fast but short-lived, as the nerve fiber do not innervate all cells of body and in cellular function need to be continuously regulated. A special kind of coordination and integration has to be provided. This function is carried out by hormones. The neural system and endocrine system jointly coordinate and regulate the physiological function in our body. 22.1 endocrine gland and hormones. Endocrine glands lack duct and hence called ductless gland. Their secretion is called hormones. The classical definition of hormone is a chemical produced by endocrine gland and released into the blood and transported to distantly located targeted organs. Has a current scientific definition as follows: hormone are non-nutrient chemical which act as intercellular messenger and produce in trace amount. The new definition covers a number of new molecules in addition to the hormones secreted by organized endocrine gland. Invertebrates possess very simple endocrine system with few hormones, whereas large number of chemicals act as hormone and provide coordination in the vertebrates. The human endocrine system is described as below. 22.2 human endocrine system. The endocrine glands and hormone producing diffused tissue cells located in different parts of the body constitute the endocrine system. Pituitary and pineal, thyroid, adrenal and pancreas and parathyroid, thymus and gonads, testes in males and ovary in females are the organized in endocrine body in our body. In addition to these, some other organs, that is gastrointestinal tract, liver, kidney, heart, also produce hormone. A brief account of the structure and function of all major endocrine glands and hypothalamus of human body is given in following section. Location of endocrine gland: here testes in males and ovary in female, pancreas, adrenal gland, thymus, parathyroid and thyroid gland. This is pineal gland, this is hypothalamus and this is pituitary gland. 22.1.1 hypothalamus. As you know, the hypothalamus is the basal part of diencephalon forebrain and it's regulate a wide spectrum of body function. It contains several group of neurosecretory cells called nuclei which produce hormones. These hormones regulate the synthesis and secretion of pituitary hormone. However, the hormones produced by hypothalamus are of two types: the releasing hormones which stimulate the secretion of pituitary hormone and inhibiting hormone which inhibit the secretion of pituitary hormone. For example, hypothalamic hormone called gonadotropin releasing hormone GnRH stimulates the pituitary synthesis and release of gonadotropins. On the other hand, somatostatin from the hypothalamus inhibits the secretion of growth hormone from the pituitary. These hormones originating in hypothalamic neuron pass through axon and release from their nerve ending. These hormones reach the pituitary gland through portal circulatory system and regulate the function of anterior pituitary. The posterior pituitary is under direct neural regulation of the hypothalamus. 22.2.2 the pituitary gland. The pituitary gland is located in bony cavity called sella turcica and is attached to the hypothalamus by its stalk. It is divided anatomically into adenohypophysis and neurohypophysis. Adenohypophysis consists of two portions, part distalis and part intermedia. The pars distalis region of pituitary, commonly called anterior pituitary, produces growth hormone, prolactin, thyroid stimulating hormone, adrenocorticotropic hormone, luteinizing hormone, and follicle stimulating hormone. Pars intermedia secrete only one hormone called melanocyte stimulating hormone. However, in human, the pars intermedia is almost merged with pars distalis. Neurohypophysis, pars nervosa, also known as posterior pituitary, store and release two hormones called oxytocin and vasopressin, which are actually synthesized by hypothalamus and transported axonally to neurohypophysis. Oversecretion of growth hormone stimulates abnormal growth of body, leading to gigantism, and low secretion of growth hormone in stunted growth, resulting in pituitary dwarfism. Excess secretion of growth hormone in adults, especially in the middle age, can result in severe disfigurement, especially of the face, called acromegaly, which may lead to serious complication and premature death if unchecked. The disease is hard to diagnose in the early stage and often goes undetected for many years until changes in external features become noticeable. Prolactin regulates the growth of mammary glands and formation of milk in them. Thyroid stimulating hormone stimulates the synthesis and secretion of thyroid hormone from thyroid gland. Adrenocorticotropic hormone stimulates the synthesis and secretion of steroid hormone called glucocorticoids from the adrenal cortex. Luteinizing hormone and follicle stimulating hormone gonadal activity enhanced called gonadotropins. In males, the luteinizing hormone stimulates synthesis and secretion of hormone called androgen from testes. In males, follicle stimulating hormone and androgen regulate spermatogenesis. In females, luteinizing hormone induces ovulation of fully matured follicles, Graafian follicles, and maintains the corpus luteum formed from the remnant of Graafian follicles. After ovulation, follicle stimulating FSH, follicle stimulating hormone, stimulates the growth and development of ovarian follicles in females. Melanocyte stimulating hormone acts on melanocytes, melanin containing cells, and regulates pigmentation of skin. Oxytocin acts on smooth muscle of our body and stimulates their contraction. In female, it stimulates the vigorous contraction of uterus at the time of childbirth and milk ejection from the mammalian gland. Vasopressin acts mainly at the kidney and stimulates reabsorption of water and electrolyte by distal tubules and thereby reduces loss of water through urine diuresis. Hence, it is also called antidiuretic hormone ADH. An impairment affecting synthesis and release of antidiuretic results in diminished ability of kidney to conserve water, leading to water loss and dehydration. This condition is known as diabetes insipidus. Here, figure 22.2, diagrammatic representation of pituitary and its relationship with hypothalamus. This is hypothalamus and these are hypothalamic neurons. This is portal circulation and here is posterior pituitary and this is anterior pituitary. 22.2.3 pineal gland. The pineal gland is located on the dorsal side of forebrain. Pineal secretes hormone melatonin. Melatonin play a very important role in the regulation of 24-hour diurnal rhythm of our body. For example, it helps in maintaining the normal rhythm of sleep-wake cycle, body temperature. In addition, melatonin also influences metabolism, pigmentation, the menstrual cycle, as well as defense capability. 22.2.4 Thyroid gland. The thyroid gland is composed of two lobes which are located on either side of the trachea. Both the lobes are interconnected with the thin flap of connective tissue called isthmus. The thyroid gland is composed of follicles and stromal tissue. Each thyroid follicle is composed of follicular cells enclosing a cavity. These follicular cells synthesize two hormones, tetraiodothyronine or thyroxine T4 and triiodothyronine T3. Iodine is essential for normal rate of hormone synthesis in the thyroid. Deficiency of iodine in our diet results in hypothyroidism and enlargement of thyroid gland, commonly called goiter. Hypothyroidism during pregnancy called defective development of maturation of growing baby, leading to stunted growth or cretinism. Mental retardation, low intelligence quotient, abnormal skin, deaf mutism, etc. In adult women, hypothyroidism may cause menstrual cycle to become irregular. Due to cancer of the body of thyroid gland, due to development of nodule in thyroid gland, the rate of synthesis and secretion of thyroid hormone is increased to abnormal high level, leading to condition called hyperthyroidism, which adversely affect our body physiology. Exothalamic goiter is a form of hyperthyroidism characterized by enlargement of thyroid gland, protrusion of eyeballs, increased basal metabolic rate, weight loss, also called Graves' disease. Thyroid hormones play an important role in regulation of basal metabolic rate. These hormones also support process of red blood cell formation. Thyroid hormone control metabolism, carbohydrate, protein, and fat. Maintenance of water electrolyte balance is also influenced by thyroid hormones. Thyroid glands also secrete proteins hormones called thyrocalcitonin, which regulate blood's calcium level. Here is diagrammatic view of position of thyroid gland and parathyroid. Ventral view. This is ventral view, and this is lateral. This is dorsal view. Thyroid, trachea, and vocal cord. This is vocal cord. This is thyroid, and this is trachea. And this is dorsal view, parathyroid gland, and this is isthmus. 2.5 parathyroid gland. In humans, 4 parathyroid glands are present on the back of the thyroid gland, one pair each in the 2 lobes of a thyroid gland. The parathyroid gland secrete a peptide hormone called parathyroid hormone PTH. This secretion of parathyroid hormone is regulated by circulating level of calcium ion. Parathyroid hormone PTH increase the calcium level in the blood. Parathyroid hormone acts on bones, stimulate the process of bone resorption, dissolution, and demineralization. Parathyroid hormone also stimulates reabsorption of calcium ion from the renal tubules and increase calcium ion absorption from digested food. It is thus clear that parathyroid hormone is hypercalcemic hormone, that is, it increases the blood calcium level. Along with thyrocalcitonin, it plays a significant role in calcium balance in our body. 22.2.6 Thymus. The thymus gland is lobular and structure located between the lungs, behind the sternum or ventral side of aorta. This thymus play a major role in development of immune system. This gland secretes the peptide hormone called thymosin. Thymosins play a major role in the differentiation of T lymphocyte, which provides cell-mediated immunity. In addition, thymosin also promotes production of antibodies to provide humoral immunity. Thymus is degenerated in old individuals, resulting in decreased production of thymosin. As the result, immune responses in old person become weak. 22.2.7 Adrenal gland. Our body has one pair of adrenal gland, one at the anterior part of each kidney. The gland is composed of 2 types of tissue. The centrally located tissue is known as adrenal medulla, and outside this lies adrenal cortex. Underproduction of hormone by adrenal cortex alters the carbohydrate metabolism, causing acute weakness and fatigue, leading to disease called Addison disease. 22.4 Diagrammatic representation of adrenal gland above kidney and B secretion by showing 2 parts of adrenal gland. This is adrenal gland and kidney. Adrenal cortex, adrenal medulla. Adrenal medulla can secrete 2 hormones called adrenaline or epinephrine and noradrenaline or norepinephrine. These are commonly called catecholamines. Adrenaline, noradrenaline are rapidly secreted in response to stresses of any kind during emergency situation and are called emergency hormone or hormones of fight or flight. These hormones increase alertness, pupillary dilation, piloerection, raising of hair, sweating, etc. Both the hormones increase the heartbeat, strength of heart contraction, and rate of respiration. Catecholamines also stimulate breakdown of glycogen, resulting in the increase of concentration of glucose in the blood. In addition, they also stimulate breakdown of lipid and protein. The adrenal cortex can be divided into three layers: zona reticularis inner layer, zona fasciculata middle layer, and zona glomerulus outer layer. The adrenal cortex secretes many hormones commonly called corticoids. The corticoids which are involved in carbohydrate metabolism are called glucocorticoids. In our body, cortisol is the main glucocorticoid. Corticoid which regulate the balance of water and electrolyte in our body is called mineralocorticoid. Aldosterone is the main mineralocorticoid in our body. Glucocorticoids stimulate gluconeogenesis, lipolysis, and proteolysis and inhibit cellular uptake and utilization of amino acids. Cortisol is also involved in maintaining the cardiovascular system as well as kidney functions. Glucocorticoids, particularly cortisol, produce anti-inflammatory reaction, suppresses the immune response. Cortisol stimulates the RBC production. Aldosterone acts mainly at the renal tubule and stimulates the reabsorption of sodium and water excretion of potassium ion and phosphate ion. Thus, aldosterone helps in maintenance of electrolyte body fluid volume, osmotic pressure, and blood pressure. A small amount of androgenic steroids are also secreted by adrenal cortex, which play a role in growth of axial hair, pubic hair, and facial hair during puberty. 22.2.8 Pancreas. Pancreas is a composite gland which acts mainly on exocrine and endocrine glands. The endocrine pancreas consists of isolate of Langerhans. There are about 1 to 2 million of isolate of Langerhans in a normal human pancreas, representing only 1 to 2% of pancreatic tissue. The two main type of cells isolate of Langerhans called alpha and beta cells. The alpha cell secrete hormones called glucagon, while beta cell secrete insulin. Glucagon is peptide hormone which play an important role in maintaining the normal blood glucose level. Glucose acts mainly on liver cell hepatocytes and stimulate glycogenolysis, resulting in an increase in blood sugar, glycemia. In addition, this hormone stimulates the process of gluconeogenesis, which also contribute to hyperglycemia. Glucagon reduces the cellular glucose uptake and utilization. Thus, glucagon hyperglycemic hormone. Insulin is a peptide hormone which play a major role in the regulation of glucose homeostasis. Insulin acts mainly on hepatocyte and adipocytes, cells of adipose tissue, and enhance cellular glucose uptake and utilization. As a result, there is a rapid movement of glucose from blood to hepatocyte, the adipocytes, resulting in decrease of blood glucose level, hypoglycemia. Insulin also stimulates secretion of glucose by glycogenesis in the target cell. The glucose homeostasis in blood is thus maintained jointly by two insulin and glucagon. Prolonged hyperglycemia led to complex disorder called diabetes mellitus, which is associated with loss of glucose through urine and formation of harmful compound known as ketone bodies. Diabetic patients are successfully treated with insulin therapy. 22.2.9 Testes. A pair of testes is present in a scrotal sac outside abdomen of the body of male individual. Testes perform dual function as primary sex organ as well as endocrine gland. Testes is composed of seminiferous tubules and stromal or interstitial fluid. The Leydig cells are interstitial cells which are present in intertubular spaces, produce a group of hormones called androgen, mainly testosterone. Androgen regulate development, maturation, and function of male accessory sex organ like epididymis, wide difference, seminal, vesicle, prostate gland, urethra, etc. These hormones stimulate muscular growth of facial and axillary hair, aggressiveness, low pitch or voice, etc. Androgen play a major stimulatory role in the process of spermatogenesis, formation of spermatozoa. Androgen acts on the central nervous system and influences the male sexual behavior, libido. These hormones produce anabolic synthetic effect on protein and carbohydrate metabolism. 22.2.10 Ovary. Female have a pair of ovary located in abdomen. Ovary is the primary female sex organ which produce one ovum during each menstrual cycle. In addition, ovary also produce two groups of steroid hormone called estrogen and progesterone. Ovary is composed of ovarian follicles and stromal tissue. The estrogen is synthesized and secreted mainly by growing ovarian follicles. After ovulation, the rupture follicle converted into a structure called corpus luteum, which secretes mainly progesterone. Estrogen produces wide-ranging effects such as stimulation of growth and activities of female secondary sex organs, development of growing ovarian follicles, appearance of female secondary sex character, high pitch voice, etc. Mammary gland development. Estrogen also regulates female sexual behavior. Progesterone support pregnancy. Progesterone also acts on the mammary gland and stimulate formation of alveoli, sac-like structure which store milk and milk secretion. 22.3 Hormones of heart, kidney and gastrointestinal tract. Now you know that endocrine glands are and their hormones. However, mentioned earlier, hormones are also secreted by some tissue which are not endocrine gland. For example, the atrial wall of our heart secrete a very important peptide hormone, atrial natriuretic factor ANF, which decreases the blood pressure. When the blood pressure is increased, ANF secreted, which causes dilation of blood vessels. This reduces the blood pressure. These juxtaglomerular cells of kidney produce peptide hormone called erythropoietin, which stimulates erythropoiesis, formation of RBC. Endocrine cells present in different parts of gastrointestinal tract secrete four major peptide hormones, mainly gastrin secretin, cholecytokinin CCK, and gastrin inhibitory peptide GIP. Gastrin acts on gastric gland and stimulates the secretion of hydrochloric acid and pepsinogen. Secretin acts on exocrine pancreas and stimulates secretion of water bicarbonates ion. CCK acts on both pancreas and gallbladder, stimulates the secretion of pancreatic enzyme bile juice, respectively. GIP inhibits the gastric secretion and motility. Several other non-endocrine tissue secrete hormones called growth factor. This factor is essential for normal growth of tissue and their repairing and regeneration. 22.4 Mechanism of hormones action. Hormones produce their effect on target tissue by binding a specific protein called hormone receptor located in target tissue only. Hormone receptor present in cell membrane of target cell are called membrane bound receptor, and receptor present inside the target cell are called intracellular receptors. Mostly nuclear receptors present in nucleus. Binding the hormone to its receptor led to the formation of hormone receptor complex. Each receptor is specific to one hormone only, and hence receptors are specific. Hormone receptor complex formation led to certain biochemical changes in target tissue. Target tissue metabolism and hence physiological function is regulated by hormones. On the basis of their chemical nature, hormones can be divided into groups. First, peptide hormone or polypeptide protein hormones. Insulin, glucagon, pituitary hormone, hypothalamic hormones, etc. Second, steroids, that is cortisol, testosterone, estradiol, and progesterone. Third, iodothyronine, thyroid hormone. 4, amino acid derivative, that is epinephrine. Hormones which interact with membrane bound receptor normally do not enter the target cell, but generates second messenger, that is cyclic MP, IP3, calcium ion, etc., which in turn regulates the cellular metabolism. That hormone which interact with intercellular receptor, that is steroid hormone, iodothyronine, etc. Mostly regulate gene expression or chromosomes function by interaction of hormone receptor complex with genome. Cumulative biochemical action resulted physiological and developmental effect. Here, protein, how protein binds Hormones related to hormones that are nature proteins. Here is receptor, ovarian membrane, first response, generation of second messenger, cyclic AMP and calcium ion. This causes biochemical response, and after that physiological response or ovarian growth shown later time. Diagrammatic represent a protein hormone. Here is action of protein hormone, how protein hormone works. And here is steroidal hormone. Steroidal hormone directly enter through plasma, uterine wall or cell membrane. Hormone estrogen. This is steroidal hormone, and this form hormone receptor complex. This is nucleus, genome, mRNA. After hormone receptor complex, this mRNA form protein. That's a physiological response. That is tissue growth and differentiation. Summary: there are special chemical which act as hormone and provide chemical coordination, integration and regulation of human body. These hormones regulate metabolism, growth and development of organs, endocrine glands or certain cells. The endocrine system composed of hypothalamus, pituitary and pineal, thyroid, adrenal, pancreas, parathyroid, thymus and gonad, testes and ovary. In addition of these, some other organs, that is gastrointestinal tract, kidney, heart, etc., also produce hormone. The pituitary gland is divided into three major parts, which are called as pars distalis, pars intermedia and pars nervosa. Pars distalis produce 6 trophic hormones. Pars intermedia secrete only 1 hormone, while pars nervosa neurohypophysis secrete 2 hormones. The pituitary hormone regulates the growth and development of somatic nervous tissue and activates the peripheral endocrine gland. Pineal gland secrete melatonin, which play a very important role in regulation. 24-hour diurnal rhythms of body rhythm, sleep state, being awake, body temperature, etc. The thyroid gland hormone play a major important role in regulation of basal metabolic rate, development of maturation of central nervous system, erythropoiesis, metabolism of carbohydrate, protein and fat, menstrual cycle. Another thyroid hormone, that is thyrocalcitonin, regulate calcium level in our body by decreasing it. The parathyroid secrete parathyroid hormone, which increase the blood calcium level and play a major role in calcium homeostasis. So it is called hypercalcemic hormone. The thymus gland secretes thymosin, which play a major role in differentiation of T lymphocytes, which provide cell-mediated immunity. In addition, thymosin also increase the production of antibodies to provide humoral immunity. The adrenal gland is composed of centrally located adrenal medulla and outer adrenal cortex. The adrenal medulla secrete epinephrine and norepinephrine. These hormones increase alertness, pupillary dilation, piloerection, sweating, heartbeat strength, heart contraction, rate of respiration, glycogenolysis, lipolysis, proteolysis. The adrenal cortex secrete glucocorticoids and mineralocorticoids. Glucocorticoids stimulate gluconeogenesis, lipolysis, proteolysis, erythropoiesis, cardiovascular system, blood pressure and glomerular filtration rate, and inhibit inflammatory reaction by suppressing the immune response. Mineralocorticoids regulate water and electrolyte content of a body. The endocrine pancreas secrete glucagon and insulin. Glucagon stimulates glycogenolysis and gluconeogenesis, resulting into hyperglycemia. Insulin stimulates cellular glucose uptake and utilization and glycogenesis, resulting in hypoglycemia. Insulin deficiency or insulin resistance result in disease called diabetes mellitus. The testes secrete androgens, which stimulate the development and maturation of and function of male accessory sex organs. Appearance of male secondary sex character, spermatogenesis, male sexual behavior, anabolic pathway, and erythropoiesis. The ovary secrete estrogen and progesterone. Estrogen stimulates growth and development of female accessory sex organs and secondary sex character. Progesterone play a major role in maintenance and pregnancy as well as mammary gland development and lactation. The atrial wall of heart produces atrial natriuretic factor (NF), which decrease the blood pressure. Kidney produce erythropoietin, which stimulates the erythropoiesis. The gastrointestinal tract secretes gastrin, secretin, and cholecytokinin and gastric inhibitory peptide. These hormones regulate the secretion of digestive juice and help in digestion. So पूरा ह्यूमन फिजियोलॉजी भाई खत्म हो गया in one lecture. Here you go, completed in one lecture. भाई साहब कुछ वन शॉट देखने की ज़रूरत नहीं, एनसीईआरटी पढ़ लो भाई, बार बार लाइन ही पढ़ो और कुछ नहीं. बच्चे झूठ मूठ में इधर वन शॉट के चक्कर में पड़े हुए हैं. भाई एनसीईआरटी पढ़ते रहो, उसके अलावा कुछ नहीं करना है यार बायो के लिए. लेक्चर देख के पूरा स्कैम किया जा रहा है साथ में. कितना लेक्चर देखो, कब तक देखो भाई, बाद में तुमको एनसीईआरटी ही पढ़ना है. तो ये सब कंप्लीट हो गया भाई अपना. चलिए भाई, बाय बाय, लव यू ऑल ब्रो. Love you all. Thank you for learning. Bye bye.